Okay, good afternoon, everybody. Welcome back. We hope your Expo and Speed Learning sessions were insightful and inspiring, and we're excited to continue with the second session of the Astronaut Scholar Technical Conference. But before we get started, I would like to bring someone up to the stage, Mr. Andrew Jones. He's an ASF alum and scholar, and he's the CEO of CARBA, and he has an important announcement for you.
Awesome. Hello, everyone. How are you guys doing? Good. I love the energy. Well, I'm Andrew Jones. I was a ASF scholar a long time ago, back in 2008. Don't do the math. I also was awarded the Neil Armstrong Award 2 years ago, which is fantastic, so that I see that in all of your futures. But I'm up here because we have internships. So we're looking for interns, and I would love to hire all ASF folk for those internships. So at CARBA, that's a company that does carbon dioxide removal. We are looking for anything and everything. So if you want to help us fight climate change and make a dent and prevent the worst effects of climate change, we would love to have you. Come find me or email me. My email is just Andrew, A-N-D-R-E-W, at CARBA, CARBA.com. And I started that company about 4 years ago, and we're on a good track so we're still an early stage company. It's a fun place to work. And then secondly, I have another company called Activated Research Company that does, we're doing early diagnostics for lung cancer through breath, so measurement technology. So if you have interest in that as well, you can just use the same email and it'll get to me. But we would love to hire you guys. You guys are the best and the brightest, and I love coming to these things every year because it gives me a lot of hope for the future, which we desperately need right now. So thank you guys for everything you do. Looking forward to this. And now it's my pleasure to introduce two very special people. First, your co-host for today, astronaut scholar and alum Ashley Monahan. Ashley is a 2018 recipient and a dedicated mentor and committee member. And second, ASF trustee and longtime supporter, and one of my friends, Carol Fitzgerald Tyler, brings a passion. Great, yeah, and Carol, she brings a passion for empowering young leaders that truly inspires us all. It was written, so it has been written. And with that, Carol, I'll kick it over to you for the first session of the ASF Technical Conference.
Thank you so much. And thank you, Andrew. Look, he's trying to hire you guys. Give him a round of applause. If you don't know what CARBA is, go look it up on the Google, as my mother says, the Google. It's a human, apparently. It's a fantastic, interesting company, and it's doing really great things for the world. So please try to engage with Andrew. That's awesome. Thank you, Andrew. So welcome back. It's 3 o'clock in the afternoon. Have you all had your coffee? Are we all ready for a very exciting session? So this is session number 2. There's my 2 guys from the U of M. Hi. How you doing? Still like my shoes? All right, good. We have good shoes today. So welcome to all of you. We are so excited to have witnessed this morning's session, which was inspiring beyond. This afternoon session, we'll be seeing some, again, phenomenal astronaut scholars. We're so excited to hear what you have to tell us. And please enjoy the session, and we will go from there. So welcome. Thank you. Thank you to all. I forgot to say this earlier. Number 1, fabulous, Ashley. Thank you. Number 2, to the people that are helping with timing and the people that are helping with microphones, thank you so much for doing that. We really appreciate your time as well. So welcome. Have some fun. Buckle in. So I'll take, and Ashley's got something to say.
Yeah, quick housekeeping. Doesn't really apply to the people currently in this room, but keep in mind that you guys were flown out here, right? We want you guys to engage and participate, so please be sure to sign in with the QR code before every event. and actually participate and enjoy it, right? Your peers are presenting really, really cool research. And we want them to have just as full of an audience as you have whenever you present. And you get that feedback, and you get to look in the audience and see, heads nodding or, curiosity in people's eyes, right? So be sure to come to events. Please attend everything. It's kind of an unwritten contract that you signed whenever you came here, right? So enjoy it, and that's how you're going to get the most benefit out of this, okay? So quick housekeeping note, just be sure to come to everything. That's it. Let's get started.
Thank you. Okay, our first presenter this afternoon is Kyle Cotre, and he's going to talk about the nerve. So welcome, Kyle.
Hello, everyone. My name is Kyle Cotre. You can see it right there. And this summer, in my lab at Vanderbilt, I was faced with a challenge. So as modern neuroscience experiments are evolving, at least in our lab, we're integrating new hardware, we're expanding tasks into 3D and virtual reality, and we need to develop faster than ever. So my job, I was hired on to build a system to make this possible. And I call this system NERV. It sounds pretty cool. So the Neuroscience Experimental Runtime for Vanderbilt is a comprehensive framework for rapid, reproducible, and hardware-synchronized neuroscience experiment design. In normal words, that means a platform to build and run game-based experiments for exploring functions of the brain. The functions my lab focused on was something called working memory. Now, working memory is best described by my professor as the ability to maintain and manipulate information in the conscious mind over a timescale of seconds. So the way that we do that in our lab is by a series of very simple games, where you'll be shown a stimulus that you have to remember, there'll be a delay with some sort of logic, and then you have to make a correct selection. Now, all of you guys have probably played this simple game before that uses that. It's called Simon, where you have to remember the color and the location that it was, and then repeat that in the correct sequence. Now, for our games, in the nerve system, our stimuli look a little different. This is called a quaddle. These are also quaddles. But for the games I'm going to be showing you guys, these are the quaddles we'll be using. So with the quaddles, and along with the nerve framework that I'm about to describe to you guys, I have created a series of games, as you can see here. Now, if it keeps playing. Now, these games are all designed in a way to evaluate a different type of working memory. But the games don't always look this cool. Most of the games before I developed Nerve look like this. A simple black background with a white stimulus, simple delays, pretty boring. It was boring to the monkeys too. That was actually a problem, where they would fall asleep during it. So the development for it is equally as boring. If you've ever used MATLAB, MATLAB is not a game development engine. So this is typically what it would look like. And it requires you have to be a professional in the psych toolbox, which is a degraded toolbox for development. On top of that, all the tools that you would need for modern neuroscience experiments would take many weeks for someone who's already in the lab and already has experience developing with MATLAB, up to 6 weeks to even get something close to experiment ready. I didn't make that up for the next slides. I asked on Slack some of my coworkers, not just trying to make it look good. But Nerve does this a little bit differently. The way to make these games is 3 simple steps. The first step is a series of form-based graphical tools where you just input the name of the game, or the name of the state. You hit checkbox if it's going to be a stimulus, a delay, a choice. And then you use 3 tools that I created. Now, these tools are responsible for automatically generating all the code that you need to get an up and running prototype in a matter of minutes. Holds all the trial logic as well. And then after that, all you have to do is press play. So this will get you an up and running task with stimuli, feedback, and scoring already. And if you need a more complicated game like the ones I'm going to show you later, all you have to do is just edit 1 script, 1 code, and everything is all condensed. Now, I think we can agree that those couple pain points that I was talking about, like the steep learning curve and the heavy code, are solved with Nerve, and that's why, like I said, it's being used at Vanderbilt right now and their adjacent medical center for human and non-human primates. Additionally, with that, Nerve is able to not only record all the behavioral log data, but also has a lot of data provenance features, such as taking screenshots of every single state that's shown, so experimenters later know what they're looking at. There's extra functions to incorporate all of our hardware that we need, like infrared-based eye tracking and reward pumps for when the monkeys get juiced and they get the correct answer. And also a pretty cool-looking experimenter screen. So the experimenter has a second monitor where they can view what's happening in real time, change attributes of the game, and conduct the experiment that way. But in order, now that we know what Nerve is and how to build games with that, we need to make sure that it's actually valid for neuroscience experiment design. So our biggest issue is making sure that when the code executes, we have a consistent delay from when the stimulus is supposed to show up and when it actually shows up on the screen. So we have a little photodiode you can see right there that's responsible for recording the exact moment those pixels flash, and then at the same time we're sending an electrical pulse to a recording system. We then take the delay of that and we look at the standard deviation of less than a millisecond of delay in that little red box with the black bold that we're interested in. And this is huge. This allows for really easy data analysis later to be able to align all the events of the game with the brain imaging data. So we're going to end this little thing with a little exercise. And for this to work, I'm going to need everybody to be on the same page. We're going to need a focus here. Everyone, for the next couple minutes, is going to be this monkey. So this is a rhesus macaque. These are the monkeys that we use in the lab. And you're going to be a special monkey because you are completing a working memory task in the nerve system to help slow cognitive decline, specifically dementia for diseases like Parkinson's and Alzheimer's. So to do that, we're going to be playing one of the 5 games here that I have created with the nerve system. But since you're monkeys, I'm going to choose for you. We're going to play the coolest one. So it's a 3D rule adaptive match. There's 3 phases to this game. You're going to be shown one of those quaddles that I was talking about in the middle of the screen. And then we're going to walk down a hallway together. Halfway down the hallway, you're going to get a flash. It'll either be blue or orange. If it's blue, when you get to the choice stage, you have to walk down the hallway that has the same quaddle you were just shown. If it's orange, you've got to walk down the opposite one. All right, are we ready? The next slide starts it. So here we go. You're going to remember this. And we're going to walk down the hallway. Now that little red cursor would be the eye position of the monkey, and then we get to the flash right about here. It's blue. So hopefully you remember what blue means. And then we get to the choice. So, yeah, I was going to say, by a show of hands, who thinks left? Who thinks right? There we go. So you'll see, as we walk down, it's going to flash green, and that would mean that all of you guys are getting a little bit of juice for your efforts. So, yeah, and just like that, you guys are very smart monkeys. Go ahead and give yourselves a round of applause for doing that. And that's all I have for you. So if you have any questions for me, email's there. If you want to play the games, you can go to nerveframework.com, or there's a QR code. So, yeah, pretty cool. But that's all. Thank you very much.
Thank you, Kyle. Way to get everybody engaged. All right, who has one question for Kyle? Andy's back. Thank you.
I'm back.
One question for Kyle. Who's got one? Thank you.
How do you explain to the monkey, like, when you see a green flash or a blue flash, like,
Yeah, so it takes a really long time. Like,
how do they figure that out?
some monkeys are smarter than others. They get it in like a couple months, but other ones it takes a lot longer. So these games, we're just implementing them now. So we have monkeys that are just now learning the games. We had simpler games. They're not playing the 3D ones yet because we're going to kind of wean them into this system because it's a lot different than the black and white than they've seen previously. So yeah. But.
Okay, now you can sit down.
Cool.
Thank you. Thanks, Kyle. All right, next we would like to welcome Nicholas Renstra, and he will be talking with us about live cell imaging. Welcome, Nicholas.
All right. Is that working? Do I just need to? Sorry, I had an AV issue. I have too many movies. I don't know how Kyle's worked. His had too many movies, but I had to plug in my own laptop, and we're making sure that is getting up there. Maybe? Hey, there we go. All right. Hi, yeah, I'm Nicholas Rienstra. I'm from the University of Wisconsin-Madison, if you can't tell by the excessive amount of red on my screen. And I'm going to talk to you guys today about some techniques that we can do a chalk talk too. Techniques that I've been developing for correlative light and electron microscopy, specifically with correlative light and electron tomography, which is a slight difference in between EM and ET. And while we're figuring this out, I'm going to explain that to you guys. EM, as you guys might know, is traditionally done with 2D micrographs, where you just basically, with tunneling electron microscopy, transmission, not tunneling, you shoot your electron through, you get a display on this detector, and you see where there were electrons and where there weren't, and you use that to determine what you were looking at. You can look at super high resolution magnification stuff, and with EM, you're typically only looking at micrographs, so you're getting 2D projections of stuff. The idea of ET is you've got a little cryo-EM grid that you have your sample on, and within your electron microscope, you have a tilt axis. So you've got this little tiny grid with your sample on it, your microscopic sample, and you're shooting electrons through it like this, and you tilt it plus and minus 60 degrees on either side. And you collect data at every single tilt angle, and you use that, you take that, and you put it through these algorithms, and you reconstruct it into these 3D models. And we use these 3D models in biology to find things that we're looking at in cells. We can use what's called subtomogram averaging to heighten the resolution of tomograms. Hey, here we go, nice. And basically, tomograms are used for looking at larger cells. Like, so I'm talking like bacteria, like viruses, that smaller, larger neurons. People can look at neurons. And the great thing about this is that you can look at bigger things than you would typically do with EM. But the problem is you have a grid. I say this grid's tiny. It's 3 millimeters. This is tiny to me. But when I'm going to do electron tomography and I'm looking at something at a magnification of 73,000 times, I have a field of view of like 500 nanometers by 500 nanometers. I have like 0.00001% of this grid in my field of view, and I don't even know what I'm doing. And that is why CLEM has been created. So the idea of CLEM is I'm not going to go into the process of like actually sample preparation of freezing it, because that's its whole thing. But once you have your sample frozen with plunge freezing, this is a typical plunge freezer that we would use in our lab. You take your sample, plunge freeze it, and then you put it into this, what I'm calling a post-vitrification fluorescence, a post-vitrification CLEM microscope. So this is a microscope, it's a light microscope that holds your samples, because I'm saying cryo-EM a lot, cryo, cold. This holds your samples at liquid nitrogen temperatures to do your sampling. Now, typically before you freeze, you would have stained these with some kind of fluorescent tags. We've talked a lot about fluorescent tags recently with everyone who's been doing microscopy here. And you take that and you look at your grid and you figure out what's on your grid sample, or what the samples are on your grid, and you find out where they are on your grid. So then once you go into electron microscopy and you're at the high resolution, you know what's going on and you know where to focus in on. Now, this is why I've been introducing this as post-vitrification CLEM, because the system that I've developed, this isn't working, okay, thank you, can you go back one? Is pre-vitrification CLEM, because I've created this device over here on the left, which I've called Fluid Cell, which is not as cool of an acronym as NERV. NERV is way better. But this is pre-vitrification CLEM, where you can take this fluorescence microscopy imaging and you can do it while you're collecting your samples in live conditions. We can honestly skip the entire next slide if we're cutting on time, but if we can move forward. Oh, I can do that now. No? Yes. Okay, if I just put my hand like this. Cool. All right. Am I doing it? Okay, here we go. I don't even know if I'm doing this, but anyway, that slide was talking about the problems with purifying samples, because if you're working with microscopic samples on macroscopic, but a tiny little grid, it's really hard to get your samples on. And a lot of our time in cryo-EM is spent perfecting our sample preparation process, from protein purification to getting your grids, to getting your proteins on the grid or your samples on the grid, your neurons, your anything, whatever you want to look at, getting it on there, freezing it, making sure the ice thickness is good, all of this. It's really hard. And once you get to the end of that sample preparation optimization process, you have, it's hard to know if you're really looking at something in its natural state, in its natural conditions. In other fields, like I'm looking at here, have a lot of different ways of creating these natural systems, natural systems of creating their samples. And one that really stood out to me are flow cells. And that's where I took this idea of connecting a flow cell and this correlative light electron microscopy system and putting it all together into this new system that we've created. And okay, so obviously, I'm just going to let this play, actually. It's not even worth battling, if it's playing. This one works now? Oh, does it? green? Wait, red. I wanted the movie. Sorry, this is awful. Is that playing? I think it is. Oh my God. It's supposed to start automatically. That's cool. Anyway, so I've created the system that is this Clem system that we have. Okay, this movie's not working then. Oh, because this switched to the PowerPoint, so none of the movies are going to work. Great, that's awesome. Anyway, okay, so basically, this is a flow cell. I seriously talk this. Okay, so we have a flow cell, and like I said, we're able to prepare our samples in natural conditions, right? Most of our samples are living in aqueous conditions, right? You're living under, and in flow is a very natural environment for this. A specific bacteria we were looking at was Calypbacter crescentus, which is what you find in like water pipes, like sticking to the sides of water pipes and stuff. So in under flow is a really natural condition to look at this and how it's developing in a cryo-EM system. So with a flow cell, obviously, as an aerospace engineer, which obviously is way changing all of what I've been doing, but we need to figure out what the flow is. So I used particle image velocimetry, which I don't know where she is, but we mentioned this earlier, particle image velocimetry, really awesome system, and I used it to determine the flow characteristics of the system. We got to wrap it up? Yeah. Anyway, okay. So I determined the flow characteristics of the system. It compares to commercial devices that do the same kind of thing but don't have cryo-EM in it. They're just normal flow cells that you can use for light microscopy. That's where the grid is. That's normal data. It looks cool. This is bright field on the left, EM on the right, more EM. Anyway, that's a tomogram. It's not going to work because the movie doesn't work. That's what's actually in it. That's Calypbacter. You can see how biofilms are developing when it's in these natural conditions under flow. And that's another image that isn't going to work. It's a 3D reconstruction from confocal microscopy that we did to show that you can really use all modalities of light microscopy with the system instead of having to be tunneled into this expensive cryo-Clem system that I showed on the first slide. And these are the people that give me money, especially Astronaut Foundation. I love you guys. Questions?
You know what Winston Churchill once said? When you're going through hell, keep going. And you did. Well done. So let's have one question for Nicholas. One question for Nicholas.
Great talk.
Yeah, that's opinion.
I probably got lost at one point, but these images you're showing up,
Yeah.
what kind of electrons are you using to get them? Are they the tunneling? Are they secondary?
So this is tunneling electron microscopy. So you've got your electrode at the top of your little vacuum tube, and you spit those out, and you funnel those with magnetic fields, and you focus those, and you blast through, and it's negative space imaging, because like you have something, if an electron isn't there with the detector, you know there is something there, and you so it's not secondary scattering, it's primary, essentially.
Perfect. Thanks for the clarification.
Yeah, of course.
Thank you so much, Nicholas. And again, you did it. Well done. Next, let's welcome Ishan Singh on optocontrol of cellular processes. Welcome, Ishan.
Okay. Oh, it's animated still. Okay. Okay. Hi. So my name's Ishan. I'm a student at Purdue, and today I want to talk to you about real-time precision optocontrol, which is a fancy way of saying I'm using light to play with cells. So my research in this lab is methods development. I'm making tools that other biologists in the lab, or biologists in general, can use to expand the toolkit that they have available to them. And when we do methods development, we have to consider what we're trying to do in cell biology. We're trying to observe and we're trying to perturb, okay? So observation, in the case of cell biology, is microscopy, as was discussed with cryo-EM. This could be with the naked eye, could be with an optical microscope, could be on a really small scale like AFM, cryo-EM, but we're trying to look at what's going on. And on the other hand, there is perturbation, which is trying to control what's going on. So it could be genetic knockout, could be chemical treatment, could be whatever. And we observe, we perturb, hopefully observe a change that tells us we did what we wanted to do. Problem is, these are separate things. We don't observe while perturbing or perturb while observing, right? We want to be able to have spatial selectivity. If I see a cell here and a cell here in an image, I want to kill this cell but not this cell. But I can't just poke and prod at different cells with my fingers, right? They're very small. So we need a way of having spatial selectivity and the ability to observe these processes in real time, hence the name real-time precision optocontrol. Now, the simplest way to implement this, which is what we've done so far, is in the confocal fluorescence microscope, which is the ubiquitous, well-commercialized system that basically all biology labs have. To understand what we've done, I first need to explain how the microscope works. So what we have is we're trying to use fluorescence to image. Fluorescence is the process by which a molecule absorbs light of one color and emits light of a different color, and we use that light to image where things are. So we have a laser at the front, or a combination of lasers. That laser goes through some combination of optics that focus down to a single point. Then any emission from that point goes back through the microscope to a detector. Now, that point is like hundreds of nanometers large, but we want to image a FOV that's microns large, hundreds of microns. And so we scan that point across the image. We do a raster scan. In particular, what's used are galvanometer mirrors, which is really small, really fast-moving mirrors. They're called galvos. These are mirrors that are controlled by the voltage that you send them. So depending on the voltage they receive, they are at a known position. Thus, any position at the grid, any position in that grid corresponds to a set of voltage values. So now say I see a cell at like the bottom left of that grid. I know the voltage values that I sent to the galvos that correspond to that cell. Then if I have a miraculous way of turning on treatment when my laser is over that cell and turning off treatment when it's not, I can do that by just determining when the galvo voltages are at that point and when the galvo voltages are not at that point. That is the whole principle of what we're doing. Perturbation occurs by having a laser activate on the sample when the scan is over the points that we want to treat. So this is what the whole system looks like. It's a little beefy, but it's the same thing that I just showed. Instead of having one laser, we now have 5 lasers. And you can see in front of 3 of those lasers, there are these devices labeled AOM. That's an acousto-optic modulator. Basically, this is a device that when I send in a voltage to it, a high voltage, it deflects the beam. And when I send in a low voltage, it doesn't deflect the beam. So if I'm sending in a high voltage, the beam is deflected into the beam path. If I'm not sending in a high voltage, the beam does not get deflected, and it's just not in the beam path at all. Thus, if I determine the voltages, or the times at which that 2D galvo scanner receives voltages that correspond to where I want to treat, I then, at the exact same time, send a high voltage into the AOMs that I want to activate. The other neat feature that we've built into this is the comparator circuit box. So rather than a priori determining where I want to treat, I can have the laser scan over, and the moment it encounters a region at which there's a high voltage, in other words, a high fluorescent signal because there's a cell there or something, that will very quickly propagate through this digital logic and then turn on the AOMs. So now we can actually have real-time treatment without interfacing with the system at all. So this is useful because light can impact protein interactions, dimerizations. It can have them release their cargo or whatever. It can impact cells, cause apoptosis, optogenetics, all sorts of things. So light-matter interaction is a very well-established thing. Our goal is basically to generalize this. So among other things, we've made it into a mosaic imaging to image huge FOVs. You can image with scripts. Here I just had fluorescent beads printed with letters, but you could have it treat cells. Image a single ROI, so if we only want to image that P for Purdue, we can only treat that spot. That's the photo bleaching that's happening right there. And finally, we need to make a software that anyone who doesn't know how to build the optics because they're a biologist can go and use. It's not that they're dumb, it's just they're a biologist. So in this software, we want to integrate all these tools that I mentioned, as well as other imaging modalities like wide field and lifetime, as well as analog modulation of the treatment laser to have various powers at the sample. Thank you to my fellow lab mates, Amy Instrumentation Facility, NIH, and especially Astronaut Scholarship Foundation. Thank you.
Thank you, Ishan. Let's have one question for Ishan.
How do you handle multiple wavelengths all going through the final system at the same time?
Yeah, so in this guy, some of the stuff I didn't mention are dichroic mirrors and other filters for specific wavelengths. But you can see we have 2 detectors. So detectors are specified for yellow light and green light, such that the fluorescent excitation from 405 and 473 goes to green, and 532 and 589 goes to the other one. We've matched up the optics very precisely such that it is as compact as possible, even though it takes like a big optical table to fit all this on. It is as compact as it can be.
Gotcha. Thank you.
Yeah.
Thank you, Ishan. Okay, next, wait for the voice. Next, we want to welcome Charles Anderson talking about the Evapinator. Welcome, Charles.
Okay, for context, the Evapinator is named after Phineas and Ferb episode, so it's not that cool. So, hi, my name is Charles Anderson. I'm an electrical engineer at Georgia Tech, and today I'm going to be presenting my work on the Evapinator, bringing universal diagnostics to the point of care. So, this story starts with the PI in my lab, Saad Bamla, who's testing malaria diagnostics in a remote Madagascar village of about 1,000 people. After a car ride and a 3-hour hike, he met Raisia, who runs a local health center that was stocked with just a single tub of medicine and diagnostics for the entire village. When we say we want to bring universal diagnostics to the point of care, what we really mean is how do we get these diagnostics into Raisia's box? But that begs the question, just what is inside this box? Well, it's essentially filled with rapid point of care tests, where with just a single drop of blood or urine, you get a result in about 10 or so minutes. So think of like pregnancy test kits, but for diseases. But these tests often have a serious issue. They're not super sensitive. So if you're not sick enough, this test may not detect it. This effect alone kills hundreds of thousands of people every single year. So how do we solve this problem? Well, there are 3 main approaches. First, you can boost sensitivity of existing tests to match lab standards. Second, you can increase connectivity, making it easier for patients to reach better, well-equipped facilities for analysis. And third, you can invent new tests that better deal with the needs of this population. And it actually turns out that dewatering or drying of samples enables all 3 of these approaches. If you remove some of the water from the sample and essentially concentrate the sample, it becomes way easier to detect targets. If you remove all of the water, the sample is now kind of stable in an inert state, and you can actually mail the sample, allowing for connectivity to better equipped facilities. And finally, it allows for new tests. So for example, instead of invasive lung tests for TB and COVID, you can essentially take patients' masks, soak them in water, shake it, and concentrate it, essentially concentrating their breath. So that's how dewatering or drying can enable all 3 of these approaches. So basically, there are solutions to this issue, but they're often super expensive, they require a lot of energy, and they're not super portable, so they're not really effective at the point of care. So in response to this, we've kind of built the Evapinator. It's a low-cost device. It can dry samples in about an hour as opposed to 12 to 48 hours for most systems. It fits into a backpack, runs off a battery pack, and costs roughly about $100 to build. So that begs the question, how did we build this device? Well, it turns out we drew a lot of inspiration from how you kind of dry clothes on a line outside. You essentially want to optimize for certain parameters. For example, you want it to kind of be windy outside, so we kind of generate a jet of air to hit the sample. You want it to be pretty sunny outside, so you want to have a heat source that heats the samples to the point where you have almost biological degradation, but not quite. And then you want to get a system of dry air. You don't really want it to be super humid or your clothes aren't really going to dry. And the way we do this is pretty simple. If you have like a cold beer bottle on a hot day, water kind of condenses on the outside of the bottle. We use a similar process to remove water from the air. And we use a Peltier cooler unit, so everything is fairly inexpensive. So putting all of this together, we have a basic system that can be used to dewater samples. Air hits the sample to remove water, water is removed from the air, and this process is essentially looped until the sample is dry. Then we add some filters to limit contamination, specifically just to the sample chamber. And finally, we add a system in order to decontaminate the system inside the sample chamber. And we use ozone to do this, which is pretty cool because we're just using ambient air to clean everything. So no consumables really needed. We additionally have a method to isolate the individual air paths so we can have multiple patient samples that are being dried or preserved at the same time. So this is kind of like a really rough look at the Evapinator. It's kind of an early prototype, but it puts everything we just talked about together. So in conclusion, we've kind of built this portable, low-cost tech that works at the point of care to preserve or dry biological samples. We're currently working with an Atlanta-based company to use this technology to preserve mRNAs for room temperature, which was a pretty big problem during COVID, and validate this technology a little bit more with mass spec. But if there are two things you take away from this presentation, I'd love for them to be the following. If you think this research applies to your own work, we're always looking for new partners to pilot our work and our technology. And if you think of any other potential applications beyond what I kind of just shared, I would love the opportunity to discuss them further. Thank you so much for your time. I wanted to thank the PI of my lab, Saad Bamla, my amazing collaborator and co-creator of the Evapinator, Rajas Purna, and the amazing help I had this summer. And thank you to the Astronaut Fellowship for giving me the chance to talk about my work. Yeah.
Thank you, Charles. How about one question for Charles?
I was wondering about the limitations of this device because I think when I was working with, or not diseases, but biomarker detection and diagnostics for stuff like glucose in breast milk or from period samples like marking for bacterial vaginosis, the clinical ranges often range from like 1 to 4 micromolars of concentration. So do you think that dry samples are able, like what is like the molecular limitation? Like can you detect something as small as glucose at that range? Do you know yet? And do you plan on doing experiments?
Yeah, that's a good question. It kind of depends on what sample you're working at. Some things can get a lot more dry than others. So for example, if you can remove all of the water and add some of the water back in, you know the exact concentration of what you're looking at. But obviously some things are a little bit more difficult than others because they do get a little bit more gummy. We know this will definitely work for something like plasma, where we can definitely detect the glucose. I don't know what the lower limit of detection, but we're currently working on that right now. Thank you.
All right, thank you so much. I just really like to say the Evapinator. All right, next we want to welcome Sun Lat, and we're going to be talking about enhancing bone healing. So welcome, Sun.
Good afternoon, everyone. My name's Sun, and I'm from the University of Central Florida. And my research focuses on improving bone healing for the use of osteoimmunomodulatory magnesium-containing nanoparticles. Now, my goal today is to not only explain what that research means, but to hopefully get you excited as a nanomedicine as much as I am. Now, to begin, we need to put our problem into context. That is, osteoporotic patients experience fractures more frequently due to a weakening of the bone microarchitecture. In fact, osteoporosis is one of the most common causes of fracture, affecting nearly 1 in 3 women and 1 in 5 men over the age of 50. Therefore, there is a critical need to find better treatments for fracture healing so that we can reduce mortality rates, reduce associated medical costs, and reduce the time that patients spend away from loved ones. Now, for us to improve bone healing, we first must understand how bone heals. Because bone healing is a complex multi-stage process that involves multiple cell types from multiple body systems. Now, the particular focus of today's talk is on the macrophage, because macrophages are important immune regulators of this bone healing response. In particular, much of the failures we see in orthopedic implants is due to the overlooking of the role of this immune system. Because in normal bone healing, there should be a timely shift from a pro-inflammatory M1 macrophage state to this anti-inflammatory M2 macrophage state. However, if there is a failure of this shift from this pro-inflammatory M1 phenotype, this can lead to chronic inflammation and therefore implant rejection. And that is why one of the more current approaches to bone healing involves osteoimmunomodulation, where we regulate this immune response to promote bone formation. That is, if we have better control over this anti-inflammatory and pro-inflammatory response of the human body, then we can have better control over bone healing overall. Now, that leads me to my project, which was to develop this osteoimmunomodulatory nanoparticle. And the particular system that we chose of nanoparticles is this family called glass ceramics, because glass ceramics have been previously shown within bone regeneration papers that they release calcium and silicon to then promote blood vessel formation and bone formation. But what has been less studied is magnesium, because magnesium by itself is known to cause anti-inflammation, but its anti-inflammatory role within bone healing has been less studied. And it conducted these experiments using hydroxyapatite as a gold standard control because it's widely used in bone healing. We used the soil gel synthesis method to create the nanoparticles, and then we used X-ray diffraction to validate our synthesis products, where we found that our nanoparticles did in fact include magnesium, silicon, and calcium. It also included, as you can see there, a glass amorphous phase and crystalline phases. This is particularly important because by changing the amount of glass and ceramic, we can thereby change the rate of degradation of our particle and ion release, and final therapeutic effect. We then used transmission electron microscopy and scanning electron microscopy confirmed that we do in fact have really cool nanoparticles. And after characterizing these nanoparticles, we then used in vitro testing with Ra2647 immortalized macrophages, with viability testing showing that as we increased the concentration between 0 and 100, there was good cell viability, but as we got to really high concentrations, we saw significant cell death. Now the question then becomes, well, if we can only work at these low concentrations, do we have any good anti-inflammatory or any inflammatory response at all at low concentrations? And the really exciting thing was yes, we did in fact find that our glass ceramic nanoparticles, compared to the gold standard hydroxyapatite nanoparticles, causes greater anti-inflammatory cell signal release. And we also found that our nanoparticle causes less pro-inflammatory cytokine release compared to hydroxyapatite at the same concentration, thereby showing that we do have a greater overall anti-inflammatory effect with these glass ceramic nanoparticles that we synthesized. This means that this is a very promising therapeutic for use in bone healing and osteoimmunomodulatory applications. Future works involve better control of this nanoparticle synthesis, so we can get better size distribution, reduce the clumping up of these nanoparticles, and importantly, one of our primary projects is incorporating these nanoparticles into dissolvable orthopedic implants so that as the bone heals, the magnesium implant dissolves and replaces the metal with bone. And that is an ongoing in vivo study. And I'd love to acknowledge my lab, all the great amazing supporters there, everyone from the Office of Prestigious Awards, Morgan and Christina for giving this opportunity, and of course, the Astronaut Scholarship Foundation for this really exciting weekend. Thank you.
Thank you, Sun. Who has a question for Sun? Oh, come on, there's got to be a question. There's one. Yay.
I was just wondering, you mentioned this a little bit towards the end with your future work with the implants, but how are you sure that there's not like these nanoparticles like diffusing out to other parts of the body and causing maybe excess bone growth? I don't know if that would be a possibility or anything like that.
Yeah, sure. So in our future in vivo studies, what we plan on doing, so what we currently do already with our magnesium implants, we've done pilot studies already, is look at the histology, or look at the slices of tissue from liver, heart, spleen, and other important vital organs, and look for the presence of magnesium or any excess tissue decay. Now, we can continue this with this project and then look further more than just, perhaps maybe more than just looking at histology, but also maybe use immunohistochemistry to see if there's any perhaps upregulation of bone-forming markers in the liver, because that would be quite bad. If you look under, if you have this paper right here, this kind of goes into more detail about the nanoparticle. And there is also a paper under my principal investigator, Dr. Razavi, where you can find more information on the project of the metal itself. And we do have one more paper that is currently in the whole submission process about this, and so you can see in the future as well, if you stay tuned.
Thank you.
Thank you so much. All right, let us now welcome Dishan Rajan from the University of Minnesota, Sunday dinner. And we're going to talk about Flow.io. Flow.io, Flow.io. Welcome, Dishan.
Hi, everyone. My name is Dilshan, and I'm going to be talking to you about the Flow.io, which is a novel interosseous needle for pediatrics. Okay, so an intraosseous needle is commonly used in pediatric emergency situations where IV access is delayed or not possible. This is quite common in pediatrics, especially because children's veins are quite small, and if you can't get an IV in about 30 seconds, they'll typically drill into the tibia right below the knee and access the vascular supply that way instead. And this occurs in about 24% of critical care procedures, so this is used quite often. However, the current devices on the market have two major problems associated with them. The first one is extravasation and infiltration, which is where the needle is drilled too far and out the other side of the bone. And this can lead to the release of harmful fluid or drug into the surrounding tissue, leading to tissue necrosis or compartment syndrome, which can be fatal in some cases. And this occurs in nearly 12% of cases and may be underreported. The other problem is associated with needle displacement, which is where the needle comes out of the body during transport because the current devices have nothing that really keeps them securely anchored in place. So based on our interviews and the problems that we identified, we came up with a need statement that pediatric ER and ICU physicians need a safer way to insert intraosseous needles because of current risks associated with fluid leakage, tissue damage, and needle displacement. To better understand this problem, we conducted a large-scale radiology study measuring over 680 pediatric CT and MRI scans. The reason that we had to use a 3D imaging modality is because the IO needle is inserted at a 45-degree angle medial, and so current X-rays, which measure anterior-posterior, just front-back, would not be enough because of the projection artifacts that may distort the measurements. So we measured the soft tissue thickness, the cortical bone thickness, and the medullary canal diameter, which are all important parameters for IO needle design. Some key findings in the study is that we found that the 15-millimeter needle, which is commonly used in infants and very young children, was too shallow in 31.6% of cases and too deep in 0.7% of cases. But what makes this a little bit more difficult is that a lot of infants have more soft tissue than slightly older kids, and so you can't always use this small needle in really small infants. And so then you'd have to use the 25-millimeter needle, which is too shallow in 1% of cases, but too deep in 35% of cases. And a lot of these cases are where they're put into infants. And this purely relies on clinical judgment because there's no really good way to figure out what needle size to use. And also the average soft tissue thickness does not linearly correlate with their age. So this led to our solution, which is theoretically a error-free version of this needle. So basically it uses the mechanical shoulder, which allows only a finite amount of the needle to actually enter into the bone. So once the threaded portion enters into the bone, then the needle will rest on the surface, and we identified the optimal needle length using the results of a radiology study. So we were able to find a needle length that works for all 680 patients in our retrospective study without being too deep or too shallow in any of them. We also added self-tapping threads to the front so that it's easier to drill in, and it also stays securely anchored in place, so it's much more difficult to remove on accident. It's still easy to remove on purpose, don't worry. So we tested this in a pediatric training bone, and this is what they use to learn how to insert IO needles. And it had worked exactly as we expected. So the needle was very easy to drill in until it reached the mechanical shoulder, at which point it required significant additional force to force it in too deep, basically. And the threads did their job as well, where it's securely locked in place, but it was still easy enough to remove by unscrewing it if you needed to. And I'd like to thank all my collaborators in ASF for this, and also it's worth noting that the development part of this was published in the ASME Journal of Medical Devices, and the radiology study portion of this was just published in Critical Care Explorations very recently, so if you guys are interested, you can read more about that. Thank you.
Thank you, Dilshan. Who has a question?
Hi, so thank you very much. This is a really interesting talk. I'm an EMT myself and have put in a few IOs in my time. I'm really just curious, well, I suppose sort of two things. First off, did you find, and you might not know this, that the self-tapping aspect of it made it easier to insert into at least the test bone? And do you know if that is more likely to cause increased pain throughout the process of the procedure?
Yeah, so it definitely does make it significantly easier to insert. So the current device has a really weird sort of like cutting head on the front that makes it quite difficult to use. So this one has 3 cutting flutes similar to like an orthopedic cannulated screw, so it's quite easy to insert. In terms of the pain, when we talk to the ICU providers, they claim that this is only like a 3 or 4 out of 10 on the pain scale, which I don't really know if that's true or not, but it looks like it would hurt. I don't know.
The insertion process doesn't really hurt that much. I think it's more about flushing fluid through that hurts.
Oh, okay. Yeah, that makes sense. Yeah, that doesn't seem like it would feel good.
Right, well, pain 3 or 4, that's too much if you ask me. But anyway, thank you so much, Dilshan. Great presentation. Next, we would love to welcome Sophia Dahlgren, and she's going to talk about drug delivery to a diseased brain. Welcome, Sophia.
Hi, I'm Sophia from the University of Washington, and today I'm talking about using polymer nanoparticles to deliver drugs to the diseased brain. So brain diseases are actually the leading cause of disability worldwide and the second leading cause of death. So the odds that a brain disease would affect you or someone you know are quite high. And these aren't things that just occur as we get older. They actually affect people at all stages of life. So starting at birth, there are diseases like hypoxic ischemic encephalopathy and cerebral palsy that will then affect people for their entire lives. And then getting into adulthood, more diseases start coming up, like multiple sclerosis, which affects motor functions. And then later in adulthood, we're seeing increasing prevalence of diseases like Parkinson's and Alzheimer's, especially with the aging population. And what makes these diseases so devastating is that when things go wrong in the brain, we just don't have very great ways of accessing and treating those diseased cells. So this is really underscoring this clinical need for more effective drug delivery to get therapeutics into the brain and offer long-term curative treatments to patients with brain diseases. So one potential solution to get more effective drug delivery is to use nanotechnology. So in my lab, we've developed a PLGA PEG nanoparticle platform, which is just a type of polymer that can effectively encapsulate drugs and get them into the brain. So importantly, we can get across the blood-brain barrier, which is responsible for keeping a lot of things out of your brain, which in general you want because you don't want just anything getting into your brain. But this also makes it very hard to treat diseases. So this is a critical barrier that we must get past. And then not only that, but once we're in the brain, these particles can penetrate deep into brain tissue, getting through the brain extracellular space. And then finally, we can actually get these particles into disease-mediating cells in the brain. So this offers us the ability to have very specific and targeted drug delivery effects. So now we just need an efficient way to actually get drugs inside of these PLGA PEG nanoparticles so that we can deliver them to the brain. So to do this, a lot of my research is focused on a method called sequential nanoprecipitation. And if you've made salad dressing, it's a very similar concept. So you're going to have oil and you're going to have water, and then you just kind of mix them together. And in the oil phase, we have our polymer and our drug that are dissolved. And once you add the water, they don't stay very soluble anymore. So first, the polymer is going to precipitate out and form really small nucleated particles of drug. And this is immediately followed by precipitation of the polymer. So to get this right, you usually have multiple organic solvents in your oil phase, and you can tune the ratios so that your drug will precipitate right before your polymer and get very high drug encapsulation efficiency inside of these nanoparticles. And then once they solidify, you can just spin them down and collect them to use in screening studies. So some of the characteristics that we look for when we're formulating these particles are a size less than 100 nanometers, and this just has to do with pore sizes in the brain. So if you want your particle to effectively diffuse through brain tissue, generally less than 100 nanometers is the size range you have to target. We're also looking for a slightly negative to near neutral surface charge so that these particles can circulate effectively through the body and not cause toxicity to cells. And then in the microscopy image we see here in green, these are the immune cells of your brain called microglia. And in red, we have our nanoparticles. And it might be hard to see from the back, but we do see colocalization of the green and the red. So this is telling us that these particles are actually getting into cells when we administer them This is for a model drug called curcumin that is loaded into these nanoparticles that you're looking at on screen. And then another thing that we monitor in our formulations is what happens to the properties of the particles after we freeze dry them. So just kind of like how you would freeze dry food to send it into outer space, we have to freeze dry our nanoparticles to send them to our collaborators in Australia. And the other solution to this problem is you just get closer collaborators, but we've gone the freeze drying route. And luckily, when we freeze dry the particles, they do maintain very good stability. So polydispersity index less than 0.2 means our particles are staying very nice and uniform. So that's what we want to see. We're also getting very high drug loading and encapsulation efficiency. So this just means the percent mass of drug in our particles is very high. And almost all of the drug that we're adding into our formulation is getting encapsulated into these particles. So overall, the PLGA-PEG platform is meeting all of these criteria for effective drug delivery to the brain. So the next steps in this work, where I'm doing a lot of my current research right now, is in adapting this PLGA-PEG platform to different drugs so we can treat a broader range of brain diseases. A lot of our work so far has focused in the neonatal brain disease space, but we're currently expanding into neurodegenerative diseases, so a lot of optimization that goes on with adapting this platform to a new drug. And then we're just testing these in model systems, so things like cells or small animals. And then lastly, the goal is to ultimately scale up these formulations and be able to get these nanotherapeutics into clinical trials so that they can actually help patients. And then I just want to quickly acknowledge my PI, Dr. Elizabeth Nance, and also lab alumna, Dr. Nuo Xu, who trained me on a lot of the formulation work and laid the foundation for this, as well as our collaborators at the University of Queensland. Thank you.
Thank you. Who has a question for Sophia? Here we go.
What methods are you using to modulate the size of your nanoparticles specifically?
Yes, so that just has to do with a lot of the formulation procedure itself. So really anything you modify about the formulation process will affect the properties, physical properties like size, surface charge, et cetera, of your particles. So you can modify the molecular weight of the polymer you're using, chain length. You can also modify what kinds of solvents you're using, and the timing of the precipitation can also affect the size of the particles that you end up with. So those are kind of the main ways that I've worked with to modulate the size. But really anything you modify about that formulation procedure will then have downstream effects on the properties of the particle you get. So there is a lot of trial and error to get everything exactly right.
Thank you so much. I will never make another salad dressing without thinking of you. All right, next, let us welcome Sean Brooks on conformations in complex carbohydrates. Welcome, Sean.
All right. Yeah, well, I hope you all are excited about chemistry, because that's what we're going to be talking about today. So I'm Sean Brooks. Yeah, that's what I'm talking about. All right, so I'm Sean Brooks. I go to the Colorado School of Mines studying chemical engineering, and I'm really excited to tell you about my research from this summer, which is looking at complex carbohydrates and the conformations of each sugar in a specific complex carbohydrate of interest, which is rhamnogalacturonin 2, or RG2. Wait, hold up. Okay, let's use the arrows. Okay. Rhamnogalacturonin 2, or RG2, is the most chemically complex carbohydrate that has been discovered in nature, and it is found in the plant cell wall of all plants. So those beautiful Texas trees you go look at, or the vegetables you may or may not have had for lunch, all contain RG2 in them. And RG2 is a particular research interest because as the most complex carbohydrate, if we can understand how to model the three-dimensional structure of it, we can create a template that can be applied to other carbohydrates. However, the problem is that experimentally, although RG2 has been of interest since the 1980s when it was first discovered, experimental progress has been relatively slow due to the fact that it can't be, the structure cannot be determined using traditional experimental techniques. So X-ray, or X-ray crystallography, which is typically used for determining things like protein structures, for those of you protein researchers in the room, cannot be applied to RG2 because it has highly flexible internal bonds that are not able to form distinct crystals. And so experimentally determining structure is very difficult. That is where computational techniques come in, like the ones that my group uses. Most of the people in my group are focused on molecular dynamic simulations of RG2, but this study that I'll be talking about today is focused on quantum mechanics simulations of each individual sugar within the RG2 molecule that you can see right here. We take each individual sugar and assess the conformational landscape, which I'll talk about in a moment. And this study, at this point in time, is limited to the 5-membered rings in RG2, but it is going to be expanded to the 6-membered rings very shortly as I continue this work. So let's do a little organic chemistry crash course real quick. So, everyone's favorite subject, right? So take both of your fingers, or both of your hands, and make an L, all right? Now, put them together like so. And congratulations, you've just made a 4-membered carbon ring. Those aren't fingers anymore. So you can move your fingers up and down and kind of twist them around without breaking your finger contact. And that shows you kind of what the conformational landscape of a carbon ring system looks like. So in this case, we're looking at 5-membered rings, not 4-membered rings. But this little wheel off to the side here shows you the 20 canonical conformations, we call those pucker states, that a 5-membered ring can adopt. Now, there are 2 major classifications of pucker states, one of which is the envelope conformation, where there are 4 atoms that are all collinear in a collinear plane, and one atom is either above or below the plane, and that is called the envelope. And the second of which is the twist conformation, in which there are 3 collinear atoms, and one sits above the plane and one sits below the plane, creating a so-called twist. And so by subjecting each 5-membered ring to all of these conformations using computational chemistry software, we can model the conformational energy landscape of the sugar as it goes through these different geometric conformations. And this is done using quantum mechanics simulations, specifically in the context of this study, density functional theory calculations. So DFT is a way to approximate the electron density of a molecule at given points in its structure. And so essentially what I was conducting for this experiment was doing geometry optimizations, which move the atoms, like perturb them very slightly within the structure, and then calculate the energy at each point in that perturbation. And then that will eventually be conducted until a local minimum is reached in the potential energy surface. And a local energy minimum indicates a stable structure. So if we go to the next slide, we can look a bit more on the computational approach that was used for these geometry optimizations. So first, we start with our RG2, beautiful molecule right here. And from there, we look at each of the 5-membered rings. That's an apiose molecule. And so first, I started by taking the base pucker state, which is just the ring, and putting it into the desired pucker state. So that right there is a 1e conformation, because that first carbon 1 is above the ring plane. And then did a little computational chemistry software development here, so we can attach 2 substituents, well, 4 substituents actually, to that ring to make the desired sugar while maintaining that base pucker state. You'll notice that it doesn't move between the 2 images. And then that was subjected to initial optimization at a low level of DFT theory to avoid the computational expense associated with higher levels of theory and just give us a basis to work off of. From this base structure, we then go down to a dihedral scan, and that's kind of like a crazy looking image, but basically all that is we have this substituent, for instance, was rotated in 15-degree increments. So this is just showing all of the different increments that were cycled through during that calculation. And a calculation was performed at each of those points to determine the energy associated with each. And then the lowest energy from those was selected in order to find the global energy minimum, and then that was subjected to a final optimization at a higher level of theory to get a more accurate picture of the energy. So now we can look at the results. These graphs there's a lot of information going on. So what I would look at is the structures on the side that I have prepared for you, where we show the lowest and highest energy pucker states for each of the 4 5-membered rings in RG2. And one thing you'll notice is that the low energy conformations for each of the sugars, the substituents tend to prefer the axial position, which are up or down relative to the ring, as opposed to the equatorial position, which is more out to the side, which you see in the high energy structures. And so this is in good correspondence with experimental results, so that's how we know our calculations are probably good so far. And then in conclusion, the furanose sugars exhibit a predictable set of low energy pucker states that vary based on the substituent location in the ring, and we were able to effectively find the global energy minimum for each sugar using these calculations. And so I'm currently extending that work to the 6-membered sugars in RG2, and yeah, that will be working on that through next year. So I'd like to thank my mentor, Vivek Bharadwaj, at the National Renewable Energy Laboratory, as well as the other members of the Computational Modeling Group, and the ASF for giving us this wonderful opportunity to talk about our work today. So with that, thank you all, and I'm open to questions.
Thank you so much, Sean. We have time for one question for Sean. Ooh.
So how do you translate from all of these individual conformation structures in these rings, and then translate from all of those individual structures into the full structure, and then optimize that? Like, what? I'm sure that's going to be a crazy computational demand to do.
Yeah, it is. That's a great question. So the theory behind that is by looking at individual rings, we can understand the thermodynamic puckering preferences for each individual sugar, and then we can compare those to the molecular dynamics simulations that the group has already conducted on the entire RG2 structure, and see how those compare. So you're right, DFT isn't going to give you a perfect understanding of the entire structure. The reason we do it on a sugar by sugar basis is because it's too large of a molecule to do geometry optimizations on entirely. So it at least gives us some indications of whether our MD is correctly parameterized. All right. Thank you.
Thank you, Sean. So for our next presenter has a wonderful name, and I hope not to break it too badly. So Tokukwu Ankwenzi. How did I do? Not bad. All right. Welcome. We're happy to have you. And we're going to talk about conversion of industrial waste gases. Welcome.
Hello. So it's Tochukwu Anaquincy. I go by TK. Make it a little easier.
Way easier. Thanks, TK.
And today we'll be discussing super equilibrium waste to hydrogen. I didn't get the memo on no intro slides, so you can enjoy a few pictures of myself here really quickly, and then just a little bit about me. But hopping right into it, not too much time today. The issue at hand that we're studying the issue to do with carbon dioxide in the atmosphere. We all know about this. Burning fossil fuels, more CO2 in the atmosphere, cause temperature to rise. We're trying to kind of tackle this issue. What we saw at the beginning of research is that a big part of the sector is due to the iron and steel plant industry. So we're tackling that part of the industry for this research. This next slide's a lot, but it'll discuss a lot of what I do. We're looking at pretty much trying to valorize waste gas from iron and steel plants. So I would encourage you all to look at, let me just put this right here. So look at this bottom right image here. This discusses what's gonna happen. Pretty much we're taking flue gas from a steel plant at high temperatures, which contains carbon dioxide, hydrogen, CO2, and CH4. And then we're converting this to blue hydrogens. That means hydrogen that we can either use back in the plant or sell off, and then carbon dioxide that we're sequestering in the ground. And this, what you'll see here is which is a lot of what I'm working on, is developing a redox catalyst for this process to get it to pretty much trying to favor a side of the equilibrium for the reaction. You can see the reaction up here is what we're trying to target, and pretty much trying to get it to stabilize on this side for this process. And on the left side of the slide goes into more of the reaction, the process we're using. So we're using something called reverse flow chemical looping. So to explain, we'll say that the biggest takeaway is that we're using the partial oxidation of our bed to drive the reaction. And let's go on. This is what our reaction setup looks like. I would urge you all to look at pretty much our analysis techniques are gas chromatography and mass spec, mostly mass spec. So that's here. This is kind of what our setup looks like with the packed bed reactor here. And again, it shows the partial oxidation that drives our reaction. And then this goes into all the nitty gritty of that. And then continuing on, this is mostly what I was working with is the catalyst that was screened for this reaction. So how I would think about it is trying to valorize this waste gas, and there's multiple steps to it. The first step is finding a catalyst that works well for it, and then from there doing scale-up procedures, testing out, see what's wrong, and then pretty much going from lab scale, pilot scale to eventually inside the plant. So again, my big thing I was working on was developing a catalyst for this. We started with Sierra oxide. That worked well for the oxidation temperatures we needed, but what somebody noticed that the peak reduction temperatures weren't exactly, they were a little too high compared to where the waste gas was in temperature-wise. It comes out of the process around 500 to 600 degrees Celsius, and we were seeing peak reduction temperatures around 800 degrees Celsius. So kind of trying to bring that peak down was what a lot of what I was working on. on the right here goes more into the synthesis technique that we're using, soil synthesis. I know somebody else uses the same type of synthesis method, actually. And yeah, and this is, by the way, this says an A right now. We haven't really published yet, so what we used aren't making that clear, but we use different dopants to actually get this to work. And this next slide goes into some of the characterization techniques. So this is our temperature program reduction and oxidation profiles from TGA. And then this is, the big thing here is showing that, again, this 560 degrees Celsius range is like where our waste is coming out. And we can see that when we have this dopant, this peak, which was used to be pretty much all like as 800 degrees Celsius range, was pushed down to more of that range we wanted to be. And we also see that we have a stable catalyst from the middle slide. We see pretty stable. And then we did ex-situ XRD to then kind of verify that we were synthesizing catalysts that we wanted to synthesize. Then going forward, we want to make sure to further optimize the catalyst for this reverse water gas shift reaction. We have a good starting point, but using other surface promoters and stuff like that. And then also we can, ooh, this did not go perfectly, but using other characterization techniques such as XPS and TM, and then going forward with that skillet procedure. And then I want to say thank you to Great State University, and then the Astronaut Scholars, and also a big shout out to my mentor, William Martin, and my PI, Dr. Fang-Sing Lee, for helping me with this research.
Thank you, TK. Who has a question for TK? Front row, I love it.
Hi, TK, how you doing? Great presentation. I have a question on the surface area calculations. Did you do any work on verifying like per area for your catalyst work in your packed bed?
Yeah, so we use specific, so we would synthesize these soldier synthesis, and then we would get it to a specific, each like particle of the catalyst would be specific surface area we use for the packed bed reactor. I'm forgetting the exact, it's like the measure is like micrometers, so forgetting the exact range it was right there, and then we'd use that, and then we'd have different experiments with different surface areas to work with. I wasn't really mentioned here too much, but we did up there.
Right, thank you.
Thank you, TK.
Yep.
We're going to pause just super briefly to make sure our AV up at the podium is working. And after that, we're going to welcome Montgomery Bodie, but let's just wait 1 second.
Okay.
Wow, look at that.
Okay. They can go back. We're good.
Okay. Thank you, Ashley. Welcome, Montgomery Bodie on diffusion generation.
Howdy. Yeah, so my name is Montgomery, and today I'm going to be talking about my work, DiffMS. So my group works on developing machine learning tools for mass spectrometry structural elucidation, and this is one of the machine learning models that we've developed. So to give an overview of sort of like the workflows that we're interested within structural elucidation and about mass spectrometry as a whole, basically a mass spectrometer at a high level is this tool that can take in some sort of samples and for all the molecules in the solution, it like hits it with a bunch of electrons, which causes it to fragment, and you get these sort of weird fragmentation patterns that you see here. But to go back from these spectra back to the original structure is a really difficult challenge. And so like an example workflow is you take a bunch of patient samples and you pass it through this mass spectrometer, and you can do some statistical analysis on like the spectra and say, oh, I know that this spectra is like strongly correlated to a certain type of disease or a certain cancer. But in order to do any sort of like real scientific discoveries, you need to figure out like what the molecule structure is. And so that's where structural elucidation comes in. You take in your spectra and you come up with like a list of proposed structures, and then you need to synthesize them and check like, does my synthesized structure actually match the original observed spectra? And this process of synthesizing and checking is extremely expensive, and it takes a long time. And so it's really important to come up with like good candidate structures like the first time through. And yeah, so there's a lot of machine learning tools that have been trying to do this, and a lot of them rely on sort of a database search where they predict some property of the molecule and then search through a known structural database. And the problem is like for metabolites and even small molecules, The space of all possible molecules is just too large, and there's like exceedingly unlikely that the molecule you're looking for has been cataloged in some node database. And so there's like a recent study that showed 87% of like real-world experimental spectra cannot be accurately matched to any known structures. So that's where DiffMS comes in. Instead of relying on a database search, what this model does is it takes the spectra and it directly generates a set of like candidate structures like conditioned on the spectra. And so for the machine learning people, this is a sort of a diffusion model, which if you've ever used like the image generation in ChatGPT, it's sort of a similar technology to that. At a high level, how this works is you start with sort of like a random sample. So you start with like a random molecule, and the model iteratively removes noise from this until it obtains like a good sample. So that's kind of what you're seeing here in this animation. And then you can do this process a bunch of times and sort of get a good understanding of the distribution of what the molecules should look like that correspond to the spectra. So we have a bunch of like experimental results, and our model outperforms all the existing machine learning tools across all metrics and like all tested data sets. Unfortunately, this like still the accuracy of getting the exact molecule is still relatively low, and so we also measure things like what percentage of the time we're able to get like a close structural match. So a good like analytical chemist is going to be able to take a set of like close matches and kind of look at this and say like, oh, like this is probably not the exact structure, but I might be able to do some like manual work and sort of get closer to the truth. So here we kind of have like some examples of this. So in the top row, we can see that our model is able to like correctly predict the true structure as the sort of the top one ranked candidate. And then in the bottom row, our model is not able to correctly reconstruct the structure. But you can see that like the top one prediction from our model and the ground truth structure differs by the placement of like one hydroxyl group. So a chemist would be able to look at this and probably figure out the true structure. And then we also provide like a bunch of analyses on like how we can continue to scale the performance of our model. So one of the problems in this space is that there's like very small amounts of like high quality labeled spectra to train a machine learning model. And so our approach sort of splits the training process into multiple stages. And so we're able to train like a much more powerful model without relying on a ton of like high quality labeled data. So very grateful to all of my amazing co-authors, as well as the funding agencies, and of course to the ASF for putting this whole event together.
Thank you. Who has a question? Oh, lots of questions. We'll take one right now.
Between y'all, so.
Very interesting. Thank you very much. Have you done any work looking into how sensitive it is to the initial conditions of the random sample that you feed into it before you begin the optimization process?
Yeah, we did, and we tested sampling from a few different distributions, and we found that it's not very sensitive to sort of the initial conditions. Yeah.
Cool, thanks.
All right. Thank you, Montgomery. Next, we would love to welcome Jasmine Franklin, talking about traumatic brain injury. Welcome, Jasmine.
Good afternoon, everybody. Wow, Laura, you're right. It is scarier up here than down there. Okay. So this is going to be about my research this summer, but first I'm going to preface with what my research group's like overall long-term goal is. So we have proteins and enzymes in our brain, right? And like Sophia told us earlier, there's a blood-brain barrier keeping all the bad things out. Well, it's also keeping the good things like those proteins and enzymes inside of our brain. And so if you find these proteins and enzymes in other parts of the body, there's probably something wrong going on. And when they're found outside of the brain, they're potential biomarkers for traumatic brain injury. And so my research this summer was just to kind of get the baseline instrument parameters for this research so we can continue forward in the future. So our goals were to compare 3 different electrodes and show if there's a significant difference in their analyzing capabilities. And hopefully, like I said, we can use these electrodes in different media to determine if we can use this technology to analyze the proteins in the body or if we have to figure out something else. So here are materials. For our analyte, we use ferroferricyanide. I know that's not a protein, but we needed something that we knew would work with electrochemistry. So we use that guy. It's just an iron cyanide compound. And then we use 3 different working electrodes. We use glassy carbon, gold, and platinum. Oh no. Yay. Okay, so the working electrodes look like this. They're about 3 millimeters long. And this part is what these 3 look like. And so this is our glassy carbon, our gold, and our platinum. Let's just give you guys an idea of what we're looking at. And then for our simple media, we use some potassium chloride or KCl. And for our complex media, we use artificial saliva. So in order to do this, we had to polish the electrodes before every single experiment. And these are my platinum electrode surfaces. So this is before we polished the platinum electrode at all. This was straight out of the package. As you can see, there's like a black film over it. And then this was after round 1 of polishing, and as you can see, there you see some of that silver peeking through, or not silver, platinum, but you know what I mean, silver color. And so we polished it over and over again until we could see more and more of that. And to get the instrument parameters for these experiments, we did a bunch of scan rate studies and electrolyte studies with these different electrodes. Scan rate studies are just changing the speed that the instrument analyzes the solution, and electrolyte studies are just changing those electrolyte concentrations. So you'd just be changing the concentrations of KCl or the artificial saliva. And then we created some calibration curves for each electrode using those data, and we changed the concentration of the analyte ferroferricyanide to get those calibration curves. And so here's a little setup. This black one, that's our working electrode. And then there's our counter electrode, that little thin wire, it's a platinum wire. And then there's our reference electrode, the glass one. And it's hooked up to a CHI instrument. And then we analyze each solution using cyclic voltammetry. Big word pretty much is a little circuit, analyze a circuit over potential, it measures the current. And here's our calibration curves. So beautiful calibration curves. And we determined that there's no significant difference, which means that we can use the most cost-effective option, which is the glassy carbon. Gold and platinum are really expensive, so we were very thankful. But this was just in the simple solution. So we had to figure out if it would work with the artificial saliva or the complex solution next. And unfortunately, we ran out of time before we can do the entire study, but we were able to run the skid rate studies with the glassy carbon, and so I'm going to show you guys the differences with the cyclic voltammograms. So this is a cyclic voltammogram. What you're looking at is this line is the oxidation, and this is the reduction of the analyte. And it's supposed to look like a duck. That's what it's supposed to look like. And so as you can see, the simple on this side looks like a beautiful duck. There's a little beak, little tail, quack, quack. But in the complex media, it looks like a goose, and that's not what we want. We want these pretty peaks so we can get nice peak separation, very small peak separation. But these wide peaks don't allow that, and then as you can see, there's a large difference in the potential. So, but we did prove that we can use this technology in more viscous and complex solutions like artificial saliva, which means we can continue forward with our research, which is super awesome. And so in conclusion, we compared 3 different working electrodes, and we decided that, hey, they're the same. We could use the cheaper one. Very cool. And we proved that we can use these electrodes in complex media so we can continue forward with our research and hopefully use this technology to identify potential biomarkers of traumatic brain injury in blood or saliva samples from actual people. And before I close, I just want to say thank you to my research advisor, Dr. Kirsten Garcia, the Citadel Chemistry Department, and these 3 lovely groups that funded this research. So Dr. Bill Porter, Schur, Boeing, and lastly to ASF for this wonderful opportunity. Thank you.
Thank you. A question for Jasmine.
Hi, I don't know how much analysis you've done on the last duck you showed, cyclic voltammetry.
The ducks.
I was interested. It kind of looks like you have a mass diffusion problem. I'd imagine the saliva, it's higher viscosity. Have you considered doing a rotating disc electrode?
No, we didn't. I actually don't know what that is.
It rotates.
Oh, no, we didn't. we
Sorry,
didn't do that. But with the saliva, we had to run it at a lot slower speeds.
Yeah.
But no, I'll bring that up. That sounds really cool. Thank you.
We also have an online question that I want to ask you.
Yes.
So this person asks, a colleague of theirs at Penn State discussed using ultrasound to open the barrier as well. Do you know anything about that, or can you comment on it?
I have no clue about any of that, but it sounds really cool.
Hey, something to look up afterwards, right?
All right, some
Okay.
follow-up actions. Thank you, Jasmine.
Thank y'all.
Okay, I just want to note that if anybody calls the Burnsville High School Chemistry Department, I was never there. Okay, you have been fantastically patient. We've had half of our conference so far this afternoon. Now, my esteemed colleague Ashley is incredibly good at waking people up and getting them out of their seats. You can't leave, but she's going to lead you in a little activity. So get ready to have a couple-minute break, and then we'll resume.
Yeah, I call this technical conference calisthenics. So, you know, we have had a lot of really great sessions, and time-wise, we don't have time to let you go, but we do have time to do thematic stretches and exercises. So everybody get up for me. Okay, we do this every year. Okay? So you're gonna repeat after me. Okay, earlier this morning, we heard from Charles about how plants fight back. So we're gonna fight back. Everybody do it with me. I don't care how stupid you look. Fight back. We're plants. We're fighting back. Okay? Okay. And then we learned about lemurs. Lemurs, right? I'm sure lemurs don't do this, but it's as much as I got. Okay? I'm sorry. I hate to insult the lemurs. Okay? Okay. Then we learned about injecting some bones. Okay? Injecting some bones. We learned about mass spec. Okay? Mass spec. Mass spec. It's like the little container, you know? It's as close as I can get, guys. Okay, we've also learned about some math. Some math. Yep. Some math. Okay? And then we've got chemistry. Okay? Like the rings. Okay, yep, it's as close as we're getting, guys. You gotta bear with me. Okay? And then finally, we've learned about cancer. Lots of cancer. Okay? They move throughout the body just like you're moving right now. Okay? And so with that, go ahead and sit back down for me and we'll keep going. Okay, thanks for tolerating my silliness. So we are going to keep going. Those of you heading out, please come back in as soon as possible. But we are going to go ahead and keep going with Eleanor O'Connor, who's going to be talking about acylated pyridines. Let's give her a hand.
Wow, well, that's a tough act to follow. But I guess I'm talking about the rings, though. So yeah, my name's Nora. I'm a chemistry major at Harvey-Mudd College, and you can kind of ignore the very complicated title of my presentation, but I'm going to talk about making new molecules. So starting off, some background on synthetic organic chemistry, which is my area of research. We really need new synthetic methods, so new ways of making molecules, in order to provide access to novel pharmaceuticals, so to make new drugs. Basically, there's a huge chemical space of molecules that we can imagine may be useful, could be pharmaceuticals, could have applications, but we don't have ways of accessing them all. So we have to develop new reactions or improve reactions that we have for better yield and efficiency. A lot of this work is peripheral editing, so if you see, oh, my bad. Which just involves changing the sides or maybe changing one functional group, adding something on to the core structure of a molecule. And we've used this to make really, really complicated molecules, but a really exciting new area of synthetic organic chemistry is skeletal editing, where we can actually change the core structure and do things like swap one atom for another or delete certain atoms. And this gives us the potential to rapidly diversify drug libraries so we can screen a lot more possible pharmaceuticals. And normally, if we wanted to change molecules in this way, we'd have to start our synthesis all the way over again with new starting materials. So this is really exciting new chemistry. Specifically, I'm working with nitrogen heterocycles, which are cyclic organic molecules with nitrogen within the ring. So you can see a ton of examples here. And the majority of small molecule drugs have nitrogen heterocycles in them, so desirable synthetic target. Previously in my lab, we've synthesized acylperols, so acyl is just that carbon with a double bond to oxygen there. And I'm trying to take these acylperols and transform them into acylpyrimidines. So we've added one nitrogen, and so now it's a 6-membered ring. And the reason we're doing this is because perols don't show up that often in small molecule drugs, but pyrimidines are the 5th most common nitrogen heterocycle. And on top of that, we see this acylated pyrimidine structure in an actual drug. So desirable synthetic target. So my work is using our green acylperol synthesis, so our nice way of synthesizing these, and combining it with some new exciting nitrogen insertion to make these acylpyrimidines. So I've hinted a bit about our green acylperol synthesis, but what's exciting about it is normally you perform coupling reactions like this. We're taking these two pretty cheap, accessible starting materials, carboxylic acids and perols, and coupling them together. But normally when you do this, the coupling agents we use, so what we use to stick them together, they're sensitizers, which means upon repeat exposure, the chemist working in the lab can basically become allergic to them and start having reactions, which is not safe and would suck for me. So we use TCFH NMI as a green coupling reagent, and we can perform this reaction in mild conditions, and we've made 19 different acylated perol products, which are all possible substrates for this nitrogen insertion. The nitrogen insertion chemistry we're borrowing from the Sharma group at the University of Oklahoma, but basically they can take perols and insert one nitrogen into either side of the double bonds, and we can get a pyrimidine. So you can ignore most of this slide, but really the important part is this blue nitrogen atom, because that's the nitrogen that inserts into the perol that we're looking at. So basically, I had to synthesize a bunch of this molecule. It's a nice yellow powder. And when you heat it up, it basically decomposes, and you get a super reactive nitrogen that just wants to jump into that perol. After synthesizing my sulfonyl-nitrene precursor, so the thing that inserts the nitrogen, I tested out a bunch of different acylated perols, and you can see I had some that worked pretty well in this green box, some present but not isolated, so they were present by mass spec, which we also just heard about. But what's really exciting about this is that all of the products boxed in green, except for the bottom left one, are completely novel molecules. We haven't seen them in the literature, so they've probably never been synthesized before. So this is really exciting. This is a very diversity-oriented synthesis because we can have the nitrogen insert on either side of the double bond. And I'm excited to keep working on this for my senior thesis next year. So in conclusion, we made these acylated pyrimidines in 2 steps from these cheap commercially available starting materials and showed that nitrogen insertion was possible on the acylperol substrates. Thanks so much to my PI, Professor David Vosberg, and my entire lab. Thanks to my funding sources and ASF for having us all here. Happy to take questions.
Thank you, Eleanor. Who has a question? Who has a question?
So like when you installing nitrogen, like how does it know where to go?
How does it know where to go? Yeah, well, so let's see if I can keep. So it's a little bit of a complicated mechanism, and I won't talk through it, but basically it wants to insert in the double bond because then it can restore aromaticity after, and it generally, we've seen some conflicting results, but generally it is preferential for it to insert away from electron withdrawing groups because a positive charge develops, and it's just more stabilizing if there's like more electrons over there, so it's like more favorable. But yeah, we see insertion on both sides.
Okay.
Thanks.
We have time for one more question. Anybody? There he is.
So on your chart where you showed what you got with the green box and the yellow box, are you able to select from these molecules, or is this a smattering of what you get when you do the reaction?
So the rows are what I would get using one acyl pyrrole, and then you get insertion on both sides of the double bond, but we're able to separate the two. So yes, you get both, and the numbers I have there are the ratios of what we get, but you're able to separate them both by column chromatography, so you can isolate them both into separate vials.
Thank you.
Okay. Thank you so much, Eleanor. All right, next we're going to welcome Annalise Hahn. And I've got your update for us. So it's extracellular vesicles. Welcome, Annalise.
Hi everyone, my name is Annalise Nguyen. I'm a senior at the University of Oklahoma, and I would tell you guys about a project I did recently over the summer using extracellular vesicles as early indicators of leptomeningeal disease. Also, I made a last-minute switch, so this presentation does not match my abstract, but now you guys get to know about 2 research projects I've done. So first, what is leptomeningeal disease? It's a devastating and aggressive complication of cancer, and it occurs when malignant cancer cells infiltrate the cerebrospinal fluid, or the CSF, and the leptomeninges, which is the membrane on the spinal cord of the brain. My PI likes to describe this as a sugar coating of the spinal cord. So you can imagine that this mere enhancement of the spinal cord may be hard to detect using standards of detection like MRI and cytology. The other phenotype is when cancer cells cluster in smaller colonies and appear inside the fluid-filled ventricles of the brain, but this is less common than the mere enhancement of the spinal cord. I also want to highlight all of the different routes in which the cancer cells can enter in the leptomeninges. So there's a wide availability of options for cancer cells to enter to. And when cancer patients are surviving longer from systemic therapies, they are developing LMD. So all cells produce extracellular vesicles, or EVs. So EVs hold a molecular cargo like mRNA, lipids, enzymes, proteins that are reflective of the parent cell. So cancer cells also release EVs. So EVs from cancer cells may hold oncogenic cargo that reflects the disease state of cancer, but it may also hold oncogenic cargo that may play a functional role as well. So EVs may play a functional role in priming a future site for tumor cell colonization. And these papers, as an example, show that EVs can hold microRNAs, growth factors like EGFR, and they all talk about how different cargo that EVs hold, they can help the cancer metastasize, which is when the cancer spreads. This is enabled by EVs carrying oncogenic material such as the mRNA transcripts for these genes here. So I have EpCAM, ECM1, PDGFRB. So all of these genes, they are of particular importance for LMD because leptomeningeal disease is a metastatic consequence. And all of these genes, they're related to cell proliferation, cell invasion, making the cells more motile. And that's all related to making the cancer cells more metastasis. So these genes in particular are relevant to LMD, but the problem is that a current analysis of EVs rely on bulk analysis. So that means current methods like ELISA or qPCR. The problem with this is that a lot of EVs are different from each other. One EV might have a different biomarker, another EV might have a different one. The problem is that in early disease detection, when you're in a low, early stages of cancer, not all of the EVs will have the disease biomarker that you're looking for. So when you take a bulk analysis, when you just take a clump of these EVs, a lot of these expressing biomarkers can just be averaged out. Therefore, I hypothesize that we can leverage the idea that these EVs may play a diagnostic role in disease progression, but having a single level analysis of these EVs will enable higher resolution sensitivity to cover the low expressing biomarkers. So this is my workflow. LMD manifests with a lot of breast, skin, adenocarcinoma cancers. So accordingly, I isolate a lot of EVs via ultrafiltration, and then that prepares the EVs for the downstream analyses, like qPCR, Western blot, ELISA. I use Western blot and nanoparticle tracking analysis specifically to validate EVs. So after the validation of my EVs with Western blot and microscopy images, I wanted to quantify the gene expression level that is detectable by qPCR. As I've mentioned, qPCR is a bulk analysis method of EVs and a way of quantifying gene expression. The green line denotes a delta CT value of 10, beyond which indicates that the detection of these gene expressions are unreliable. And as you can see in most of these charts here, 4 or 5 of them, you can see that the delta CT value is above 10, indicating the unreliable detection of these target genes, because at higher CT values, you're reaching the detection limit of the qPCR assay. And this is the same case for the human cell lines. Consistently across most of the different genes, the RNA detection becomes unreliable with the bulk analysis. The hope is that with the single EV analysis, we can have a better level of detection. Essentially, the way it works is that single EVs are tethered onto a microchip via antibody binding, and from there we can probe for the oncogenic RNA and protein in a single EV level. This enhances detection level as evidenced by this previous paper, who used the same sort of a device that exceeded detection limit beyond qPCR and ELISA by 3 orders of magnitude. So before probing for my genes of interest, I want to test with VEGF as a control because VEGF is a well-characterized vascular marker in MEF cells. The green denotes detection of VEGF mRNA, and the red indicates VEGF protein, with each dot denoting a single EV. So confirming the co-localization of VEGF mRNA and protein on a single EV level. Moving ahead, I've designed the RNA detection probe for EpCAM and plan to implement this in the next stages of my project. EpCAM specifically is implicated in LMD progression and is actually used as a biomarker for circulating tumor cells. The future directions include comparing the sensitivities of the single EV biochip to the bulk analysis, so qPCR and ELISA, and also investigating EV heterogeneity and establishing an LMD model as well. I'm especially interested in using EVs as a way to implicate LMD in the CSF area. And I want to thank the Dr. Kim and Dr. Jang lab groups at the University of Texas MD Anderson Cancer Center, of course the ASF for giving me the platform to speak today, and the funding and the CPRIT Research Training Award as well. Thank you.
Thank you very much. Who has a question for Annalise?
I have a question. Great presentation. I was curious, so I know you're looking at different genetic markers and for their heterogeneity. I was wondering if you've noticed any heterogeneity in the metabolism of the EVs at all?
Do you mean like, sorry, can you clarify the metabolisms of the EVs, like what they're
Just
holding?
like different like processes that they're going through, or if they experience or it like accelerate different like co-metabolites or anything like that.
Yeah, I think that would be, I haven't explored that. So far I'm exploring just like what they're holding and how they relate to LMD, but I think that could be really achieved in like a functional model, as I mentioned, an LMD model for like a rat or mouse model to see how they actually play a role in developing LMD in the CSF area. Thank you.
Thank you so much. Thank you, Annalise. Okay, next we're going to welcome Benjamin Raufman, designing novel superatomic structures. Welcome, Benjamin.
All right. Good afternoon, everyone. My name is Benjamin. I'm a rising senior at the University of Maryland, College Park. For the past 5.5 years, I've been working at the David Haas Nanoscience Lab up the street at Towson University, primarily investigating superatomic structures of gold. Back to the computer. So before I begin, I'd like to make a few acknowledgments. So first and foremost is Dr. David Haas for making all of the work I'm about to show possible, as well as Daryush, a fellow undergraduate, and Catherine and Dylan, who are actually high school students who were very helpful to me in completing this work. And in that picture, you'll actually see a fellow astronaut scholar, Wilson Turner, who invited me to the lab in the first place. So to begin, I'll give a bit of an introduction to gold nanoclusters. So when we're talking about nanoscience, we're talking about structures that have at least one of their dimensions in the nanoscale region. For a lot of the nanoparticles we've been talking about today, if we're talking about lipid nanoparticles, glass nanoparticles, everything that was mentioned earlier, we're talking about a range of sizes. So you can get a range of diameters. Typically, you'll show electron micrographs that have structures of all different sizes. But when we get to below 2 nanometers, we start to see something special happen, and that's we see discrete molecular structures. And to differentiate these between larger particles, scientists started calling these nanoclusters. And the reason why these specific sizes arise is because of something, a framework that was developed by physicists actually called superatom theory, where we're saying that the electrons of the valence electrons of the metal atoms in the cluster are delocalizing fully over the cluster shell, and they're reforming into orbitals that resemble very closely to those of elements. So we call these pseudo s orbitals, pseudo p orbitals. And we get stable structures, stable molecular structures when they are completely filled, similar to noble gases. So up here on the right, you'll see the AU13 structure that shows up a lot. And this is an 8-electron structure, very similar to noble gases. And we're interested in these structures because they really are very exciting for applications ranging from catalysis to drug delivery to bioimaging, because they're frequently photoluminescent. And they have a very straightforward synthesis. So it's pretty exciting for a synthetic chemist because we can do a lot of different things with them and get a lot of different results. So the main structure that I'm actually interested in is this AU11 cluster that's capped by that ligand in the bottom right called triphenylphosphine. And the reason I'm excited about it is because A, it's a very effective catalyst. It's been shown to be effective in several different catalytic mechanisms. B, it's super easy to synthesize. If you mix the reagents necessary to get this cluster in almost any ratio, you're going to end up with it in some amount. It can also assemble into larger structures, which I'll get into in a little bit. But unfortunately, it's non-luminescent, and it's a little bit lacking in stability. So I was interested in, can we replace some of these gold atoms with heterometals to modulate these properties, which is an effect that's been seen in several other cluster systems. So after several months of work and quite a bit of effort, I managed to actually synthesize a platinum-doped cluster. We saw a platinum atom selectively replace the central gold atom of the AU11 cluster. Here is a X-ray crystallographic representation where we're seeing C3V symmetry for any of the inorganic chemists in the audience. And of course, we were able to unlock photoluminescence from the cluster, and this was pretty exciting. Otherwise, I wouldn't be telling you about it today. You can see that the gold 11 cluster, the black line, you see absolutely no photoluminescence, whereas the doped cluster, we're seeing some almost a 2% quantum yield, which is pretty much a measure of how bright it is. So the next question I asked is, okay, well, why is this happening? So the physical structure, as I told you earlier, didn't really, or I should mention that the physical structure didn't really change much from the gold 11. The structure that I showed you before from the X-ray crystallography is almost exactly the same as that of gold 11. So we looked into the electronic structures, and the primary method we did this is first through absorption spectroscopy, where we're looking at how it interacts with light. And again, it doesn't really look that different from the gold 11, a slight redshift of the major peaks. However, when we look at, on the right, this is a voltammogram, we see that there's a large expansion of the HOMO-LUMO gap. So the number that I included up there is the measure of the energy distance between the lowest unoccupied, or the lowest occupied molecular orbital, the highest occupied molecular orbital, and the lowest unoccupied molecular orbital. And we're seeing that's a humongous expansion, and this has previously been correlated in other studies to modulate the photoluminescence. So I was pretty excited by this. I then wanted to see, okay, well, we got this expansion. Can we do anything else that would improve our photoluminescence? So I replaced the bromide ligands on the cluster with iodide ligands. I thought this would be a fun synthetic challenge. This took a few weeks of effort as well, and I actually saw a reduced photoluminescence. I didn't really understand why. The electronics look pretty similar to the bromide version. However, I did notice that this was likely due to something called the heavy atom effect, which I won't really get into now. But this got me thinking that if we can use the ligand to change the photoluminescence, then how can we improve the photoluminescence? So if you work with luminescence at all, you probably know that the primary method by which we modulate this is through either rigidifying a molecule or loosening the molecule, because the two primary methods by which an excited state can relax is either through the molecule wiggling a little bit or a photon being emitted. So I more recently developed a synthesis with what's called a biphosphine ligand, where we now have two binding sites to the outside of our cluster, which should force the conformation to maintain in a tighter constriction, a tighter state, which can accelerate the radiative process of emitting a photon. And you can see that we have almost a 5 times enhancement in our photoluminescence. So this was all very exciting, but you might be wondering, so where are we actually going with this? A lot of what I talked about today is also being done in work with quantum dots, which recently won a Nobel Prize. Maybe nanoclusters will be next. But what is special about nanoclusters and the superatom concept is that we can sort of use these structures as what we call artificial elements and sort of build up to the three-dimensional state, a three-dimensional periodic table where we have these clusters that we can really tune the properties of and then assemble into these larger structures. So what we're really excited about and what we're working towards now is building these larger structures that you can see on the left and trying to develop them, these tunable units, into novel materials. Yeah. So thank you very much to the ASF and everyone for listening, and I'll take any questions.
Thank you so much. We have time for one super quick question.
Okay, I'll make this quick. I'm not a chemist, so I won't be asking any deep questions anyway. So you mentioned these like super materials that could be made with these atoms. Could you project any potential properties that they may have that are advantageous to current materials? And if so, what applications might they have?
So unfortunately, I am not an engineer. I don't work in a lot of the application work. But I will tell you that one of the most exciting ones is, if I go back a slide, you'll see that linear expansion of the AU13 units, AU11 units, whatever. One of the things that's really exciting about this is they're trying to use it for something called nanowires, where they're trying to develop novel electronics using very long linear expansions. And I included 3 units up there. I think recent work being done has expanded that far, and they have novel like polymerization style reactions to do this. So a lot of that is in electronics and other things at the nanoscale. Yeah.
All right, thank you, Benjamin.
Thank you very much.
So many clusters, so little time. All right, let us now welcome Charles Fiorilio, and I hope I did that right. Since I'm half Italian, I should have. Modeling the photoelectron. Welcome, Charles.
I think you got the full Italian pronunciation. So, hi everyone. I'm Charlie. I'm from WashU, and I'm going to talk today about theoretical models for photodetachment. Yeah. So, what is photodetachment? Now we're going even smaller than the nanoscale down to diatomics is mostly what I study. Think back to your intro science class. You've got a photon. Photon comes in, hits a molecule. If it has enough energy, it'll eject an electron with whatever's left over. The benefit of doing photoelectron spectroscopy is that in comparison to a photon, an electron has a wave function, so you can detect it on a 2D detector, and you can generate a picture of what that electron wave function looks like, and that's related to the initial electronic structure, any dynamics that are happening here, and that's what we call a photoelectron angular distribution. So that's kind of what my theory goes off of. Why is this a process we want to understand? Well, to start kind of big picture, anything in quantum mechanics, you can't see what's going on, so experimentally, you know what you start with, you know what you measure, but you don't know what's going on in the middle from experiments. So you need theory. Theory is kind of you get to put in what's happening in the middle, and you can piece it all together. Photodetachment is a good process to both be able to predict theoretically because it states that easily get rid of electrons and states that kind of our resonances in electron transfer are important energy transfer. But it's also a great opportunity to test our kind of quantum theory, basics of quantum theory, because it's such a simple but also really sensitive process because we're trying to predict a whole 2D spectra. So it gives us a chance to test our electronic structure treatments of small molecules, to test how we're treating that outgoing electron, and those are kind of light matter interactions and charged particle in a field are common areas through all of science. So what can we actually test? Like what are our inputs into the calculations? Well, it's really a simple process. We just have the molecule. It's just the initial state. This is our the transition operator. So that's basically the light hits it, what's happening, and that's analytics. So we know that fully. Final molecule, and then the electron. So we really are just worried about how we're treating the molecule, how we're treating the outgoing electron. Within that, there are different things that we can do. With the molecule, we know the process is very sensitive to the electronic structure of the anion, the electronic structure that the electron is coming from. So we can use that as a test to say, all right, in detachment, do we think it's coming from the state that it's actually coming from? Is it direct detachment? Is it hitting anything in the process? We can put in resonances and see, okay, maybe it's going up to this unknown state and coming off. And then kind of in the vein of getting better theory for all quantum processes, we can say, for this molecule, for this system, are we using a high enough level of theory to actually capture the electronics of the molecule. And then for the electron, it's common you see here just a general plane wave. And so we can see, okay, does a plane wave work? Do we get good results? If so, that's good to know for kind of future calculations. With this molecule, we know it doesn't generate a field when it interacts with light. But kind of some of the interesting cases and what I've been working on recently is methods to describe that outgoing electron if the molecule produces a strong field. So let's say kind of the stuff I've been working on, this is a single center approach. This is clearly a diatomic, does not have a single center. And so how can we describe a molecule under the kind of by nature the influence of a two-center field? And we can do that through using a different coordinate system, a two-center coordinate system. So all this kind of practice it in photo detachment, and it has all these kind of cool applications that go through all of scattering theory and all of continuum dynamics, which are important throughout physics and are how we understand small particles. So that's kind of a brief overview. If you want to talk math or anything, please come to me. It's a lot of fun. I would like to thank the MABS Lab and the ASF and all of you guys for listening. If you have any questions, I'd love to take them.
Who has one question for Charlie?
Hi. Are you trying like perturbative calculations, or are you trying to do numerical?
Everything is numerical. The electronic structures are generated through, like, we use both HF, but a couple cluster calculations to get correlation. And then all the cross sections are also numerical calculations because you're dealing with molecular orbitals, so you can't do any angular momentum algebra or anything.
Thank you.
All right. Thank you, Charlie. All right. Next, we would like to welcome Colton Bruni, adding spatiotemporal information. Spatial. Thank you.
Thank you.
Boy, aren't I doing great this afternoon.
Yeah,
Welcome, Colton.
I have to follow up on the guy who just said quantum mechanics was simple, so.
Yeah, exactly. Welcome, Colton.
Anyway, hello, I am Colton Bruni. I'm a rising senior in chemical engineering at LSU. And today I'll be discussing adding spatial temporal information to the autoencoder radiation anomaly detection. I'm going to throw another acronym at you. I'm just going to call it ARAD from now on. All right. So first off, what type of radiation we're working on? I know today we've discussed a lot of different types of radiation, so let's discuss specifically what I'm going to be working on, which is high energy photons coming out of radioactive sources. So shown here on this plot, I have you have a source which is emitting high energy photons at specific emission lines, which are then counted by a detector, counted by their energy. However, if you notice, it's not clean lines, right? You have this broad spectrum around these peaks. This is because the detector physics leads to a Gaussian distribution around the emission lines. In addition to this, to make things a little more complicated for longer term measurements, the actual rate at which the photons are emitted follow a Poisson distribution. So that is exactly what I'm hoping to look at. And so now I'm going to go into an example of a type of spectrum that we're going to be looking at. So out of these two, one of them is a dangerous isotope that we would like to be able to detect, an anomaly per se. And the other one is a standard isotope that is known, that is kind of pervasive in our environment. So if anyone wants to take a guess, any guesses on which one's the bad one? Left, right? Right one? Left one? Okay. Well, sorry for the left. That is potassium-40, which is a natural part of potassium. Not all potassium is potassium-40. That would be really bad if it was. And it's also commonly found in bananas. The one on the right, for all those who guessed that, that is weapons-grade plutonium. So our algorithm's trying to get better than that. Now, the point I'm trying to illustrate here is that background can significantly contribute to the full spectrum. Traditionally, if you were just to have a straight-up plutonium source, you would see much higher counts in the proper emission lines. However, because this is actually a shielded source, I didn't tell you that, so it's kind of hard for y'all. A lot of background noise is contributing to this, so it makes it very difficult. Now try to imagine doing that 40 times a second. That's the point of these algorithms that we're trying to develop. Okay. So the use case for this, because we actually have algorithms that are pretty good at this already, but we want to make an algorithm that can work in a urban environment. So specifically, let's say you have a truck with a detector on it going around a city to make sure we can find any medical isotopes that are important, or industrial isotopes, or like plutonium or something bad like that. And so what characterizes these encounters is that they're faster, they have more noise, moving vehicle, so the spectrum you get is wildly different background, right? So we need to make an algorithm that can handle all these conditions while also alerting the alarms and then not alarming for anything else. Okay, so the way ARAD works is that it takes in a input spectrum and then it goes through an encoder, which is a layer, several convolutional layers, decodes it down to a latent space, so it's 128 size down to about 816 size. And this learns abstract qualities similar to what Montgomery was talking about. And then after this, we then do an extra step where we decode to a reconstructed graph. So it basically tries to recreate. Now, the way this really works is that it works on reconstruction losses. So you see at the top here is a background spectrum, and you notice the red line, which is our reconstruct, and our original, which is the blue, are right on top of each other. Okay, this is working properly. It's able to detect, it's able to mimic it, so it knows that it's not an anomaly. However, whenever the red line and the blue line don't match, that means it doesn't understand what it's seeing, and therefore it's likely an anomaly. I think that's cobalt 60. Yeah, but what's spatial temporal? What does that mean? That just means over time, okay? So what we're trying to do is not only have one spectrum being inputted, but multiple at a time. If we can do multiple at a time, bring all these into a latent space, and then add another step of a long short-term memory node, it can learn to deal with noise and other background, more complicated background spectrum over time. So I would like to thank LSU College of Engineering and my lab, the Intersectional AI and Security Lab with Dr. James Gwally, and of course the Astronaut Scholarship Foundation.
Great. Who has a question for Colton?
He's smiling. What you got, Britton?
Hey, great talk. Where did you get weapons-grade plutonium to measure for this project?
I'm not at liberty to say. That was your
Get it
question,
at the
man?
weapons-grade plutonium store.
Yeah, Amazon does not ship that.
One more question.
Oh, thank you. Okay, good.
I'll take a question if that's okay. Oh, sorry. Is there any room for wavelet analysis or like time domain, just other Fourier type analyses in these, or?
The problem is there's the noise patterns in it, and also the emission lines and like where they're distributed. I suppose you could do that, but it wouldn't work over time. The noise is too bad. The Poisson distribution, it doesn't come out as nice. You should see some of these spectrums, they're pretty bad. I could talk more about that later. That's interesting.
Thanks.
Sounds a riveting dinner conversation. Thank you so much, Colton. All right, I'm going to try this my best. Next, we are going to welcome Jiwan Chen on tissue repair. I did it? Oh, yay me. Thank you. Welcome, Jiwan.
So volumetric muscle loss leads to devastating and permanent muscle damage in patients and currently lacks effective treatment options. Hello everyone, I'm Zhiyuan Chen, and today I'm excited to share some of my work at the Tazi Lab at Purdue University on programmable biodegradation of injectable hydrogels for tissue repair.
Stand a little closer to the mic for me.
Gotcha.
Thank you.
So since we're the ASF innovators, I'm going to talk about astronauts first. So here's astronaut Alexander, and many of you may be familiar with the effects of microgravity and muscle atrophy or muscle loss in astronauts, and this was partially combated with special diets and exercises. However, volumetric muscle loss, on the other hand, which can arise from traumatic injuries or surgical procedures that leads to loss of significant muscle volume, is clinically complicated and currently lacks effective treatment options. So volumetric muscle loss, or VML, refers to extensive loss of muscle tissue so significant that it leads to scar tissue formation formation that's non-functional and leads to permanent disability. And we don't want that. So we need to fill in that gap with new cells. To do so, we need to incorporate a bioscaffolding, much like the scaffolding you see on the right at construction sites. Sorry, this is not working for me. Much like you see as the scaffolding at construction sites on the right. And to do so, we need to incorporate novel biomaterials to fill in that gap and to support cell infiltration. Now, when you think about biomaterials, it usually means metallic implants, such as the hip implants in the picture to the left. However, the biomaterials we want for VML scaffolding need to be dynamic and durable based on tissue infiltration and can degrade away as new tissue takes its place. So now imagine a soft and water-rich material, much like Jello, called hydrogel, being used to build that scaffolding and deliver healing tools for cells to infiltrate that wound site. Here we present a suite of programmable biodegradable hydrogels for tissue engineering purposes. And hydrogels have a wide array of properties that's significant to cell infiltration and cell proliferation. And hydrogel injectability is one of such properties that's critical in clinical applications where time and ease of use is of the essence. Here, the video on the right is an excellent demonstration of shear flow, where solid hydrogen particles in the syringe flows through the needle and into the delivery site almost as a liquid and then returns to a solid form. And mechanical testing on the left also supports this, where you can see the moduli crosses over at higher strain levels. Now, this is accomplished with granular hydrogels known as microspherical hydrogels, referred to as microgels, as you can see in the image below. Now, microgels are fabricated with advanced microfluidics to form microgels of 120 micrometers in diameter, about the width of your human hair. And these are formed with the aqueous or water-based polymer flow in the central inlet, along with oil inlets on the sides to form water and oil droplets, and that's later processed with UV light. And we've done extensive degradation testing on these hydrogels, both in vitro and in vivo with animal models. In vitro degradation test shows that these hydrogels can degrade from days to months. And in vivo work with animal studies, as you can see, also supports this, where injection site 1 with the slower degradation polymers and the injection site 4 has the fastest degrading polymers, and the latter nearly matches background signal in about 10 days. Here's another in vivo testing with histology sections, and in the white you can see cell nuclei, and red and green are hydrogels. So on the left, red hydrogels have degraded, and showing evidence of cell infiltration with the white nuclei stains. On the right, however, the red hydrogels did not degrade, and there's less evidence of cell infiltration. So this is promising, and current and future work involves the extensive and rigorous animal studies to investigate the degradation of these hydrogels and how it links with cell infiltration. And there's also application in the time-dependent release of drugs. We want to eventually demonstrate that these hydrogels can work in humans for tissue engineering applications. So today we went over the challenges of volumetric muscle loss, hydrogels as dynamic biomaterials for VML scaffolding, making microgels with full-focusing microfluidic devices and their shear flow properties, how hydrogel degradation links with in vivo cell infiltration, and how that can help support tissue engineering and helping volumetric muscle loss no longer be a permanent disability for patients. I'd like to thank the Astronaut Scholarship Foundation at Purdue University, each of its scholarship offices, research offices, the NIH, Purdue Imaging Facility for help with data collection, as well as members of the Mayo Micromaterials Lab for their support. Thank you so much for listening, and any questions are welcome.
Thank you so much. Who has a question?
Hello. So you had mentioned injecting this hydrogel for future use in, I guess, medicine, but I was just curious about, like, if you guys anticipate any sort of like immune response, kind of like with graft versus host disease, or, I mean, do you anticipate that? Is there anything that you guys have done to kind of correct for that, or anything that you think is interesting about that sort of thing?
Yeah, totally. So immune response, some of the ways we're combating the body's immune response and starting can be aided with small molecule delivery with this degradable hydrogels. The hydrogel polymer material itself is a polysaccharide, carbohydrate, and a variation of it is commonly used in plastic surgery, cosmetic medicine. So we know that it's not cytotoxic and it doesn't lead to any adverse immune response in the body when you inject it.
Awesome, thank you.
Thanks. Another question?
Great presentation. So one question was, what do you use to give it its shear-thing properties? Did you say you incorporated microparticles into it?
So the hydrogels themselves are microspherical hydrogels. They are about 120 micrometers in diameter. So with this granular hydrogels, when you push upon it, as you can see in the previous material testing, it acts almost as a liquid and flows through the needle. And once it's outside of the needle, it returns to its solid form again.
All right. And my quick follow-up question is, so in that case, on your synthesis map, how did you determine like what was the, I guess, ideal shear thinning amount you wanted?
Yeah, so the video that I demonstrated is done with a 27-gauge syringe needle. So that's usually what we use to inject insulin. Usually we draw a bit lower gauge or wider diameter, so 24 gauge, 21 gauge for that. And the pressure is just the right amount for your hand to push upon it for surgeons to inject a biomaterial.
Thank you so much, Siwan. Next, we would like to welcome Jason Lee, efficiency droop contributors. If you want to know about drooping, let me know. Welcome, Jason.
If I had a nickel for every time I had this comment, I'd be a very rich man. All right, thank you for the lovely introduction. So today I'm going to be talking about efficiency droop contributors in indium gallium nitride green LEDs. And so as we're combating climate change, we're moving towards carbon neutrality, obviously we want to be making electric devices both more efficient and use less energy. And so one such device is solid-state lighting or LED-based lighting, and it can be up to 90% more efficient than more traditional lighting sources such as incandescence and fluorescence. And so the current way we produce white light is that we have a blue LED, which is the most efficient LED, and we coat it with something called a phosphor, and this down converts the blue light into yellow light, which looks like white light to our eyes. And this has been a great advancement in terms of lighting. The inventors of the blue LED won this cute little award, awarded by the Swedish people, named after a dynamite guy, if you may have heard of it. But despite the illustrious award awarded to blue LEDs, it still suffers from some problems, namely efficiency losses due to this down conversion process. And so a more efficient way to go about this is to use color mixed LEDs. And so this is where you put red, blue, and green LEDs together on the same device, and this can increase efficiency by up to 60%. However, the problem with this is that green LEDs kind of suck. And so they suffer from efficiency droop, and this is when the efficiency of the device drops dramatically at higher currents and higher optical powers, which is where you want to be operating the device in an industrial setting. And so the DOE estimates that we can save up to 196 million metric tons of carbon dioxide annually if we meet their efficiency goals by 2035. And as a reference, this is about 3% of total US emissions currently. And I always joke that 3% is not that much, but if a couple people all do 3%, we solve the problem, right? And so just kind of a little more context on exactly what this efficiency droop is. And so this is efficiency curve of the LED device that we tested. And so as you can see, the LED increases with current density, and you reach a maximum efficiency point, and then it kind of decreases. And this is the droop. And so if we had a device with no droop, it would simply reach the maximum efficiency point and just flatline. However, losses are physical, and so this is a modeled perfect device with material limitations limited by physics. However, this is what we actually measure out of the device. And so essentially my research is to figure out what happens. We lose over 40% of the efficiency, and this is not that great. And so in terms of why this is important, so if you notice, the efficiency at low currents is actually not that bad. You get about 60% efficiency out of the device. However, this kind of current regime you're operating in is where you operate a lot of very low power electronics, and so stuff like LED indicators, cell phone displays, flashlights. And it's in this kind of high current regime where you get a lot of efficiency loss, where you actually operate things in industrial settings, so stuff like lighting, car headlamps, lighting for horticulture. And lighting accounts for over 8.5% of total electricity in the US, and so we will decrease our electricity consumption both nationally and per household if we improve LEDs. And so next, I'll go over the results, and I'll skip over the methodology just because I don't think it's very interesting. If you want an overview, I essentially just press buttons and stuff happens. And sometimes, most of the time, things go wrong. And sometimes I get good data, and it's like black magic. And then I look happy, kind of like the monkey that we saw in an earlier presentation. I'm like, so this is the curve that we saw earlier. And so this is the kind of perfect curve without any of the different efficiency loss mechanisms that I'll shortly describe. And so, this is no droop. And so we get kind of about a 49% relative droop contribution from something called inherent Auger-Mettner recombination. And I think a lot of these will be just words to y'all, so I'll kind of skim over them. And then we have polarization-induced effects. And so this is about 35% of the droop. We have thermal effects, which is about 17%. And then finally, this isn't a droop contributor, but we also have light extraction losses, where a lot of the photons emitted by the LED is trapped inside the device. And so essentially, in conclusion, we conclude that in order to make green LEDs better, we want to create materials that essentially mitigates these two effects because they account for a vast majority of where the loss is happening. And so I don't really have a cute acknowledgement slides with my PI, and so I'll just say thank you to Professor John Byram. He's the GOAT, and you can quote me on that. Thank you for my parents and my sister who are here for their undying support, and thank you for the Astronaut Scholarship Foundation for flying me out here to yap about green LEDs. All right, thank you, and I'll take any questions.
Nicely done. Welcome, Mom, Dad, and sister. How great to be here to support Jason. Who has a question for Jason?
So you mentioned that this research was on the indium gallium nitride LEDs. Do you see similar proportions of the droop contributors depending on the different materials for green LEDs? Yeah,
no, great question. And so, at least for this preliminary research where we kind of made this framework of how exactly to decouple these droop contributors, we're mainly looking at green devices because this is the main motivation for visible light. But next year, we're going to try to look at kind of LEDs across the visible spectrum and kind of create a roadmap and look at exactly where this efficiency loss is happening at each material system. And so to our understanding, we think that because of the higher indium content, in order to produce this green light in GaN specifically, you end up having more polarization-induced losses. And then we think that in, let's say, red LEDs, which are in the kind of aluminum, gallium, indium nitride system, there probably is more thermal losses. And so it definitely depends on the exact material that you're using to produce this light.
Great. One more question for Jason.
Are these LEDs that you're testing, are they commercially available, or are you making them in-house? And if you are making them in-house, if you could give a 20-second explanation of your fabrication process.
Okay, yes, so I don't have to give the 20-second rundown because they are commercial, but I think that, so they're Cree surface-mounted LEDs, but I think that in the future it would be very interesting to actually grow your own devices, and I don't think many of you are very familiar with the way that LEDs are structured, but essentially we think that based on exactly the way you structure the LED, that some of the proportions may differ, and so that would be a very interesting study to see, but probably above my pay grade, and so we'll wait for a grad student to do it. All right, thank you all once again.
Thank you, Jason. Okay, let us now welcome Peyton Hay, Electromagnetic Metasurfaces. Welcome, Peyton.
All right, I really hope the microphones don't pick up the noises coming from my stomach right now. So, but I'm Peyton Hay. I'm a senior at the Florida Institute of Technology. I study electrical engineering. And this summer I had the great opportunity of spending 10 weeks at the University of Michigan in the radiation laboratory studying electromagnetic metasurfaces. And we're not going to jump right into that. We're going to go back a little bit and look at a wider view of the topic first. So we'll look at what are electromagnetic waves. These are coupled and oscillating electric and magnetic fields. This is what they look like. They go together and they propagate through space. And these are associated, or really they are, in wireless communications, anything wireless and even light itself. And in the field of applied electromagnetics, we are after the control of these electromagnetic waves. And unfortunately for us, control of these waves is central to many parts of our daily lives. We can look at medical imaging, MRIs, CTs, X-rays, of course, wireless, telecommunications, your cellular devices, Wi-Fi, Bluetooth, et cetera, and then remote sensing, and that's how we can observe things about our Earth and even out at other planets. But the way these devices control electromagnetic waves, there's a problem with that, and the reason is because the way we're doing that is we're looking at the macroscopic geometry. What I mean by this is, if you take an example of like a dish TV antenna, what we're doing is we have a curved surface, and waves come down, hit the curved surface, and that's how we direct them into the antenna hanging on the front. Another example would be a lens, light going through a lens. We have a curved surface through some material, and the light focuses at a certain point, or certain distance from the lens. And lastly, here's another example, it's an antenna. We add these metal elements more and more to the front of this to focus the beam or the energy of this antenna to make it more focused in the long term. And this comes with some trade-offs and limitations. These can be bulky, as you can see, and depending on the material, they can be heavy. They can also be inefficient, especially when we try to combat the size and weight limitations, and also difficult and costly to fabricate, especially when we're trying to be more precise. Now, the research I do is in metasurfaces. These are artificial 2D structures. They're planar, and they're composed of these tiny elements that we design called meta-atoms. And the approach here is, instead of looking at the macroscopic geometry, what we're doing is we're tailoring the relative microscopic geometry of a material, or really creating a material, to get these same effects from the waves. So this right here, this is just an example of a metasurface. Each of these gold crosses is a meta-atom, and all meta-atoms really have a few of the same characteristics. One, they achieve full power transfer, so we don't want any wasted energy. Two, they want to achieve control. That's really what we're after, and you can control polarization, phase, and or amplitude. And then last but not least, they have to be sub-wavelength in size. And this is really important. This is where the relative microscopic geometry comes into play, because you can see them. You can clearly see them on the screen. But it's microscopic to the wave that we're trying to interact with. And it's really the same way that you look at the walls, you look at this podium, and we see that it's wood. We don't see that, the atoms that it's actually made up of. And this is what we want to do for the wave itself. This gives us a lot of good benefits. One, they can be low profile, just millimeters in thickness on these sheets. Very lightweight, efficient because we're designing them at the meta-atom level, and then easy and cheap to fabricate because we can use techniques that we already use, like printed circuit board fabrication or nanofabrication. This is an example of one way you can design these meta-atoms. It's a 3-sheet design. Electrical engineers love this because you can basically model it with a simple circuit, and we like circuits. So this is one meta-atom. You can put them together. Here is more of a one-dimensional sheet, and you can design each layer and each meta-atom so that they have that small effect on the wave that contributes to the overall effect. So here we have a few examples. On the left, we've created a lens. These are just computer simulations. So we'll have waves, planar waves coming up from the bottom, and then when they hit the surface, they will focus, just like you would see in a lens. And if we take the same lens next to this and we kind of do it backwards, where we have a cylindrical wave on top that propagates outward, like if you dropped a rock in a pond, and it hits this surface, then they'll collimate. That's exactly what happens. And then a separate surface on the right, it achieves wide-angle refraction. Basically, it changes the direction of the wave. So the waves will come down here in this example, and then they will go 70 degrees in that direction. And this is with full power transfer, no losses. All right, here's one practical example. Of course, I'm going to make it space-based. This is a CubeSat. I don't have a lot of time, but this is a 3U. And basically, this reflector array is a type of metasurface that was put on here. And it's basically achieves the same effects as that parabolic dish. And we've made it so that it can fold around 3 sides of the CubeSat. And it increased the data rates from just under 10 kilobits per second to over 100 megabits per second, because you've put effectively this very large antenna on this small CubeSat. And I know this is one example, but really overall, metasurfaces are promising alternatives to traditional electromagnetic waves. I'll run through this real quick. Future work, reconfigurable intelligent surfaces. These can be tuned and adapted. You can put these on buildings, help with 6G technologies. And then conformal metasurfaces, which is really what I'm interested in, is shaping these. Instead of making them planar, you can shape them around surfaces that can't be changed, like the nose cone of an aircraft. Real quick, I'd like to acknowledge the Astronaut Scholarship Foundation for bringing us out here, all of us, and it's been a pleasure meeting you all. The support I had at the University of Michigan for hosting me for 10 weeks, and then of course the faculty and staff mentors I have at Florida Institute of Technology. And with that, I'll take your questions.
Thank you, Peyton. Who has a question?
So you mentioned briefly that this is also something you can do with like lenses and visible light.
Yeah.
So, like, what is the smallest wavelength or range that you guys have been able to do this kind of thing with?
I think we have achieved the visible light stuff. I think Harvard's actually working on that. That's very, very small. And when we start getting, we can do this up into the terahertz frequencies, which are visible lights up there. But yeah, the limitation I think we start running into is the fabrication there. But I don't know of any specific visible light examples yet, but it's being worked on.
Another question for Peyton.
So how does this differ from like a phased array or a Fresnel lens type approach? I know they're kind of two different things.
Yeah, I'm not sure about the Fresnel lens, but the phased array, so in a phased array we have multiple antennas that each have a phase shifter associated with them. And we feed them out of phase. It's just like, I'm going to use the pond example again. If we drop multiple rocks in a pond all at the same time, well then they'll all go, the resulting wave will go straight. But if you take two rocks and you drop them out of, at different times, you'll see that the constructive interference goes off in one direction. And that's what happens with a phased array. With this, we can design this to happen at the meta-atom level. We don't need phase shifters because those eat up a lot of energy and they're bulky and they're heavy. So you can design the material to achieve that same effect without all the negatives. And with the tunable surfaces, you can steer beams, you can make them wider or more narrow, or even create multiple beams with this. Yeah.
All right, thank you so much. I'm still consumed with the phrase, always combating size and weight. Yes, indeed. That was funny. I know it's 5:30, but that was funny. All right, here's the deal. We're a little bit ahead of schedule, and our last presenter today is remote, so we have a couple minutes. I want everybody to stand up and have a quick stretch. Levez-vous, as they say in French. Oui? Quick stretch. Quick stretch. We're in the home stretch, so stretch a little bit. All right, good stretch. Have a seat. We have 3 more fantastic presenters. Have a seat, please. Heaven forbid we should get out early. What? All right. Have a seat. Thanks so much. All right, next, and I've had a preview of this presentation, so super excited to introduce Britton Perdue on optical master clock. Go Britton.
So, good evening, everybody. My name is Britton Perdue. I'm an electrical engineer from Mississippi State University. And to start things off, I'd like to give a big thank you to the Astronaut Scholarship Foundation for having me up here to do this talk. And I'd like to say that this work is unclassified and it has been cleared for dissemination by the U.S. Naval Observatory. Now, with that said, my talk today is going to be about time atomic clocks and how we package up time at the time factory and then send it out all over the country to your phones and wherever, and GPS and wherever it needs to go. But the first question is, why should you even care about time? Why does it matter to any of us? If I had to bet, everyone in this room has checked their phone or their watch or something at least 10 times today, maybe 10 times since this session started. And I'd further go on to bet that probably not a single one of you has taken a pause to think about where that information actually comes from. And that's actually really important, because precise time is the underpinning of many, many critical infrastructures across the United States and across the globe. I've got a bit of an example to kind of bring that to light. Who gets or responds to emails as like part of their job? Who in here, like most people, right? Okay, now take a second and imagine they didn't have timestamps on them. Just didn't have them. You had to guess, you had no idea. Is that report done yet? I'm leaving early today. You have no idea when it happens. That'd be kind of annoying, maybe even make it impossible to do some aspects of your job. And now you take problems like that and you scale them up to the scale of infrastructure, to the scale of GPS, power plants, entire countries. And you realize that if you don't have good timing, you've actually got pretty big problems. And furthermore, infrastructure and the entire world really is growing increasingly more interconnected, increasingly more technical, and as such, the time standards that they're relying on are growing increasingly tight. So what do we do to keep timing current with the rest of technology as it progresses? Does anyone know what we're looking at here? I know we've got some physics people in the world, in the room. Any guesses? Laser? There are some lasers in there. What that is a cloud of strontium atoms being held at a couple of microkelvin. That is a couple of hundred of thousandths of a degree Kelvin. But more interesting than that, to me, is what it does. What it does is act as the most stable pendulum, the most stable tick-tock that humanity has ever discovered. And this is a strontium optical atomic clock, and it's an optical atomic clock, which is kind of the next generation. It's the hot new technology in timing. It's been around for about 20 years, actually, and it's slated to give between a 100 and about a 10,000 times performance increase over previous microwave standards. So actually, if you remember the first slide from Peyton's talk, where it was like the little waves, the little wiggles, basically, we've made the wiggles a lot faster, and that is fundamentally better for timing. But that also introduces a problem, because these clocks tick too fast to count. That's one of two problems. And the second problem is they rely on fundamentally different physics, which means that we don't really understand how to package up the time and send it out to the world. So, first of all, let's talk about that second one a little more, which is how do you package it up? What you need to do in order to distribute the time, in order for all of these systems, these infrastructures to leverage reliable time information, you can't just use one clock. You have to take multiple, and then you have to average them down, turn it into one really, really good, ultra-stable timing output, and then you can send that to the world. And that's what GPS is running on, provided by yours truly at the Naval Observatory, and that's what your phones and your computers and your power plants get. Now, our question is, how do you do that with optical atomic clocks? So we built a fun little system, and it's going to solve really both of these problems, or it's going to investigate both of these problems, which is, how do you measure it? They're really hard to measure because they tick too fast. And then also, how do you integrate it into the timescale? So first of all, our system takes relative measurements. Instead of taking direct measurements using this really expensive hardware we can use, it takes relative measurements. And then the second thing is we actually, just with relative measurements, are able to extract out the performance of each of the individual clocks. This is a variance basically showing how noisy, how bad of an oscillator your clock is. Lower is better, etc. So we were able to proof out both of those two concepts in just a 10-week program, and it's really promising work for how my former team at the Naval Observatory is going to make this operational. With that, thank you to everyone who has helped me with this work. Once again, thank you to the Astronaut Scholarship Foundation, and I'm here for questions.
Thank you, Britton. It's about time to ask him some questions. See what I did there? It's about time. Who's got a question? Go ahead.
Okay. This might be a dumb question. So if your clock is going faster than basically what our normal time scale is, you're saying like the little wiggle, you're speeding that up, that's hertz, right? So that's 1 per second.
Yes.
How is that gonna change if it's not per second? Like how is your timing scale changing?
Okay, great. So actually, all of our time scales are basically down converted. So time signals operate at about 10 megahertz in general, and then your electronics can turn that into like 1 hertz and increment your clocks. Really, the issue is, even our old clocks are operating on like a couple hundred, I think, gigahertz. But the issue is that they're still slow enough to turn into that 10 megahertz signal and then to turn into that like 1 hertz, 1 tick-tock per second signal. Now we're operating on like 200 terahertz, 400 terahertz. It's way faster, and we have to use very specialized equipment to actually scale it down to something without basically exploding the noise. So that's kind of what we're working with there.
Thank you. One more question?
What isotope of strontium are you using? And like, is it pure 90? Is it 90?
Oh gosh, I don't know. I don't actually work with strontium clocks. That's just the best. It's the best example. We have 2 groups at USNO. So there's a strontium group, and what strontium is really good at is the very short time. But the actual clocks they use for timescales, that they're hoping to put into the timescales, are calcium atomic clocks.
And are those like pure isotope? I'd imagine they might have.
Yes, I think they are enhanced isotopes. I'm on the optics and electrical engineering side, so I don't really deal with that.
Sorry, I had to throw something nuclear at you, my bad.
Right.
And we have one more question. It's not really a question, but I can't not ask you this from online, from Cassius. Tell us the story about your car breaking down outside of Kamala Harris's house.
Okay, so thanks, Cash. So the U.S. Naval Observatory is where all of this wonderful science gets done. It's also where the vice president lives. And so I was there 2 summers ago in D.C. when I was leaving work, and I think like an engine orientation sensor in my 23-year-old car failed. And so it rolled to a stop right in the middle of Massachusetts Avenue. And let me tell you, I could not count to 5 before there were 3 Secret Service vans very eager to help me figure out whatever was wrong with my car. So anyway.
Was that your fun fact?
No, actually.
Wow.
I completely forgot about that story.
There you go. Which reminder, turn in your fun facts to Lisa. Okay, they're due before lunch tomorrow. That's a good segue. Okay, awesome job.
Thank you.
Thank you, Britton. We're very eager to help you move your vehicle. All right. Annalene Whitson, welcome, and we're going to talk about rooftop units. Welcome, Annalene.
Hi, everybody. I'm Annalene Whitson. I study electrical engineering at Texas A&M University, and this summer I was at the National Renewable Energy Laboratory, and I was working on modeling HVAC systems, so what cools and heats our buildings. So to start with, why do we care about this? Well, commercial buildings report that 40% of their energy consumption goes towards heating, ventilation, and cooling across the United States. So if you add all of that up, it adds up to a lot of energy that we're spending on this. And commercial buildings report that between 2009 and 2019, they saw a 10-fold increase in the amount of their energy that comes from DC distributed energy resources. And so what this means is buildings are typically supplied from the power grid, but we see an increase in things like on-site solar panels and batteries as we integrate renewable energy and other backup energy sources, and these provide DC power instead of AC power. And so it's important to understand the impact that this will have on how our buildings function, because we'd all be upset if we switched power sources, and all of a sudden it was 10 degrees hotter in here. And so to understand this impact, we first have to understand rooftop units, which are a popular type of commercial HVAC unit, and they look like what you see on that building there. And they include all the components you need to heat and cool air before it's sent into a building. And so my job was to create a model of this, so we're gonna abstract out a little bit. And we start with a power source that supplies an electrical system made of controls, heaters, motor drives, and motors. And then this supplies energy to a thermal system made up of a heat pump, which then controls the zone temperature or our building temperature. And specifically, we think that when we supply these systems from a DC source instead of an AC source, we'll see a difference because of the rectification step, where the AC input power is rectified into DC power, and then it has to be inverted back into AC power. So it's kind of a redundant step, but it's necessary for controls because we want the frequency and amplitude of the input power to our controls and motors to be what we desire it to be. And so if we start with DC power instead of starting with AC power, we can skip the rectification step, and we can avoid switching losses and conduction losses that occur during that process. And so what this means is I think that if we use DC distributed energy resources instead of always using the power grid, we'll see a difference in HVAC systems' coefficient of performance, which is provided by manufacturers, and it represents the ratio of thermal energy to electrical energy. And it's a way of us estimating how efficient these systems are. But this has been done based on using AC power sources and not DC power sources. And so it's not information we have readily available to us when we want to understand what future implementation looks like. And so that's where my model will come in, where we'll do a more detailed component level model of a rooftop unit. And then from these more detailed calculations, we'll be able to back out this coefficient of performance that gives us the general idea of how the system is working. And so what the flow of information looks like for this is we start with the knowledge of what the electrical energy consumption of the AC system or the system supplied by AC power needs to be, because we know the coefficient of performance in this case. And then my model will calculate the instantaneous electrical power being consumed and the output instantaneous mechanical power. Then we want to match that mechanical power to be the same in the case where we supply the system from DC power, because we want the same thermal performance of the system. And then we can calculate things in the opposite direction to understand what electrical energy consumption needs to occur in order to achieve the same heating and cooling of our commercial buildings. And so when we put all of this into MATLAB and we simulate it over a 24-hour period, we get this idea where a thermal model requests that the system turn on and off as heating and cooling is necessary. And you can see each peak as the system turns on to provide hot or cold air to the building. And at each of these points, we saw the result we expected, which was that the case where the system was supplied by DC power consumed less average power than the case where the system was supplied by AC power. And so this is promising because it means that we can avoid predictable losses if we use alternative energy sources that are already being deployed anyway. Additionally, my model allows us to look at the dynamics that happen on more of an electrical time scale over seconds or microseconds. And so this would allow us to understand other important electrical parameters, such as the power factor and the rise time, as you're constantly turning the system on and off. And so that's a case for potential future work with this. And then we also did some validation of the model where we looked at if we were accurately predicting the zone temperature based on the requested temperature. And we found that when we incorporated these more dynamic electrical models, we still got accurate results, which means that, and we actually improved our accuracy a little bit because it includes things like the power rating of the equipment that we're using. And so what this showed us is that although there's future work to be done, maybe with some of these smaller timescale dynamics, this is promising for the fact that distributed energy resources have the possibility to create huge energy savings across the United States and improve the sustainability of the equipment we use in the US. And I'd like to say thank you to Omkar Gatpande, who's my mentor at NREL, as well as Priya Darshan, who I worked with. And thank you to ASF for the opportunity to present and this weekend.
Thank you, Annalene. Who has a question?
Can you expand a bit more on why the efficiency of the system differs between DC and AC? Is it something like not needing to use a rectifier or more complicated than that?
It's primarily from not needing to use a rectifier. Honestly, I would say for now that like the level of complexity of my model, that would be what was covered. There's the possibility for energy savings with controls as well, but I didn't get into the level of like modeling the efficiency of the controls. So in this process, it would just be rectification. That would be the efficiency savings.
Another question. Anybody? Bueller? Bueller?
Sort of a random question, but I noticed on your graphs that you had measured for 24 hours in Denver. And I was kind of just curious why you measured in Denver, why is it sourced in Denver, that sort of thing.
The internship was in Denver, so the National Renewable Energy Lab is in Denver.
All right, thank you so much, Annalene. So now, due to the fantastic wonders of technology, we are, a drum roll please, we are welcoming Alexis Spence remotely. Alexis, we're so happy to have you virtually. Welcome.
I'm so happy to be here. I am currently still at my internship in California, so I'm really happy to be able to talk to you guys about the research that I've been able to do for the last 8 or 9 weeks. So like you said, my name is Alexis Spence. I'm a junior at North Carolina Agricultural and Technical State University, majoring in physics with a minor in professional theater acting. And today I will be talking about dark matter and some dark matter searches I've done here at SLAC, the National Accelerator Laboratory at Stanford University. But first, a little bit about what I'm going to talk about today. So first, what is matter? Why do we know nothing about it? Second, what we're looking for, and we're looking for more matter. And third, what we're going to do if we find it. So first, we're going to start with that first question. So if you aren't in the astronomy community or astrophysics community, you may not be aware that there was a new telescope recently saw first light down in Chile called the Vera Rubin Observatory. Now, this telescope is amazing. It is a technological behemoth and has the largest digital camera ever built here at SLAC in California that is taking images of the universe, large-scale images of the universe and its complexities and galaxies at a rate of billions of pixels a second. Now, just for some reference, your phone camera and pictures that large HD TVs can handle is about 4,000. This camera is taking pixels in 4 billion per picture. 4 billion pictures with a B. 4 billion pixels, excuse me. So this is a picture from that recent release of images from the telescope. And as you can see here, there's a lot of galaxies that are filled with stars and dust and matter. So you see, we can see all of this matter. However, there's a lot that we also can't see that isn't necessarily matter. Now, that is what's most interesting. You see, everything that is matter that we interact with day to day, what we are made up of, what the stars are made of, you can find on the standard model. Now, if you're in physics or chemistry, you're very familiar with the standard model. It's what makes up atoms. It's that make up molecules, that make up everything that we interact with. However, this standard model only makes up about 16% of everything in the universe, matter-wise. 16%. The other 84% is made up of dark matter. Now, dark matter is incredibly important as it has a lot of control over the universe and its structures, including the structure of the large-scale universe, of how galaxies are formed and orbit around each other, and including how our own evolution came to be. Now, dark matter is difficult because we don't know much about it, and that means a lot of our theories are very widespread across many different fields, across different disciplines, and here's a diagram of some of those theories that we have. Now, with those theories, as there are so many, there's also a very wide range of which we are searching through. So this is a diagram of the sizes of matter that we are looking for to find any dark matter candidates. Now, because these go from smaller than than a millimeter, all the way to the size of 30 masses the size of the sun. There's a lot to look through. So we have a lot of different methods of detection. Some of those methods include indirect detection, which uses satellites looking at the large-scale structure of the universe, usually galaxies and supernovas and how they bend around each other. There's collider production, which is exactly how it sounds, trying to make dark matter particles from colliding ordinary particles together, and direct detection. My group here at SLAC works on that third one. So for direct detection, my group uses superconducting detectors, which are thermally conductive materials lowered to millikelvin temperatures to access superconducting properties of those materials. Fractional millikelvin temperatures are colder than the vacuum of space. So vacuum of space gets to about 2-3 Kelvin. These conductors, we get down to 0.15 Kelvin. Incredibly cold. So we use these detectors like a big thermometer. When a dark matter particle has an interaction with an ordinary particle, it will leave deposited energy on our detectors. We are looking for this deposited energy as evidence of dark matter interacting with ordinary matter in the space of this detector. As I mentioned before, we have to get these detectors down to millikelvin temperatures. So we use dilution, cryogenic fridges, to lower those temperatures. Each one of the levels of that fridge, you can see there's like rings in each level down, goes down to a different temperature. So that top ring is just a vacuum chamber, and then it goes down to about 50 Kelvin, down to about 4 Kelvin, and then all the way to those fractional Kelvin temperatures that we're looking for. So then our superconductor, like a thermometer, looks for that energy or those interactions to leave energy and therefore heat on our superconductors. For a little more technical explanation, when energy is left on our superconductors, the resistance in the conductor rises, and therefore the current lowers. That drop in current is what our detectors are looking for specifically to therefore find that dark matter deposited energy on our conductors. So we make superconducting circuits, we stick them in these cryogenic dilution fridges, and we take a lot of measurements. We recently just closed our fridge here at SLAC, and we are currently in the process of taking those data measurements of dark matter searching through a bunch of different instruments, all attached to the bottom of our dilution fridge. That is a lot of work. So have we found anything? Yes, actually we have. We have found really good evidence for dark matter, which is why we continue to search. Those first evidence of dark matter was through those large-scale galactic structures that we see in the universe, principally discovered by Miss Vera Rubin, the namesake of that new telescope that opened up in Chile. So we found those evidence of dark matter in a lot of different scales, from the way galaxies rotate all the way to the cosmic microwave background radiation we see everywhere in the universe. And we have started to find some smaller bits of evidence of dark matter on our detectors. However, we have found new background noise that is preventing us from finding specific kinds of dark matter candidates. We call this background noise LEE. My research is currently trying to characterize that LEE, but as I said, we are currently in the process of collecting data, and therefore I don't have much to characterize yet, but we will get there eventually. So what are we going to do if we find dark matter? Like I said, we have, but it is only a very small portion of dark matter that we've discovered. So I relate this to how we used to know about the electromagnetic spectrum. As we all know, the electromagnetic spectrum is vast. And what we can see, what we normally use, is that visual part of the spectrum with our eyes. And we knew a lot about how the electromagnetic spectrum could be split in different colors. That was originally discovered by Newton. However, over time, technology has developed, and we have found more uses for the vast different parts of the spectrum, all the way from radio waves, then microwaves, infrared, X-rays, gamma rays, and we've had all these new technological developments because of it. We could do the same with dark matter. If we were able to find these vast new kinds of matter, we could access completely different parts of the universe. We could understand better our evolution and how we came to be here on Earth, how galaxies were created, how they currently rotate and move, and the simple existence of our universe itself. as I said, a majority of our universe is dark matter. If you include dark energy, which is a whole other headache for the cosmological and astrophysics fields, there is a lot we don't know about the universe and a lot we have to discover and understand. So finally, what we know about dark matter is that it makes up about 85% of the universe. We have a lot of theories about it and numerous experiments around the world, from here to CERN over in Switzerland and Canada and all around the world. And we have a lot of goals to find it. And when we do find it, it will change the way that we understand technology and the universe itself. I would like to thank, obviously, the Astronaut Scholarship Foundation for really encouraging me to present. I'm always proud to present. It is one of my favorite things, and I love science communication, along with the DimQuest group, who I'm working with here at SLAC, along with my mentors, Dr. Kelly Stifter and Dr. Aditi Pradeep, along with the entire DimQuest group. They have been an absolutely amazing group of people, and I'm really, really proud of the science that they do here. Thank you.
Awesome. Thank you. Dark matter matters. Let's have a question for Alexis. There's one in the way back. Oh, there we go.
Do you have an expected interaction rate for dark matter with regular matter?
We don't. Currently, as I said, a lot of these searches are pretty new. We are dealing more with interference at the moment. Dark matter has proved to be incredibly elusive, and we have not found any kind of consistent rates of finding dark matter. As I mentioned, LEE, low energy excess, is a current background noise that I'm dealing with characterizing. And it's incredibly difficult because there are so many different parameters that we have to worry about: the electromagnetic spectrum, neutrinos from the sun, vibrational interference, temperature interference. There are a lot of other issues that we're trying to find and figure out before we're really able to narrow down the rate at which we could find dark matter.
Good. Thank you. One more question for Alexis.
So what's your favorite dark matter candidate? Are you looking for WIMPs, or are you kind of branching out looking for other stuff as well?
Yeah, so our group specifically works in WIMPs. It is a really prominent theory of dark matter at the moment. A lot of different groups here, and we have a very large collaboration with Snow Lab up in Canada that also looks for WIMPs. However, there are some theories that are coming a little more into popularity, such as freeze-in dark matter. I'm not even going to try to explain that theory. A lot of this is very elusive and has to do with quantum ideas. But yes, WIMPs is the strongest and most popular theory of dark matter at the moment.
Dark matter matters. Thank you so much, Alexis. We wish you were here, but we loved your presentation.
Thank you so much.
And by the way, as a veteran of working at Stanford, head up to the Rosewood Hotel for a drink in the Redwood Room. Just saying. Not that I was ever there, maybe 500 times. Good. Thank you all. Well, we are spot on time. Thanks to the co-pilot over here who keeps everything on time. Get it? Time. It's time to say thank you to Ashley. So a couple of things. First of all, thank you to Ashley, but also I want to give a big thank you to Janelle and the entire ASF team. What a day we have had. Thank you so much. Before we close off, a couple of housekeeping things. So your wonderful scholars, your fun facts need to be to Janelle and or Lisa by the end of lunch tomorrow. When do they have to go? And when is tomorrow? And what day is tomorrow? 16th Saturday. Very good. Janelle or Lisa, be sure you get them there by lunch tomorrow. And if, don't anybody ask me for any fun facts. No, 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 no. So What an outstanding afternoon. These presentations are a huge and powerful reminder of the future that we are all building together. You have shown us your curiosity, you've shown us your ingenuity, you've shown us your bold vision and how you are leading the way for all of humanity. We've talked about drooping, we've talked about repaired tissues, and we've witnessed the birth of the evaporator. So you are all incredible. Thank you to every one of our presenters. Thank you to those who helped prepare you. Thank you for all of the attendees, families, friends, distinguished guests. Thank you so much. As a quick reminder, we are back here tomorrow at 9 a.m. When? There we go. And what day? There we go. It'll be the third and final technical conference session. And for tonight, we would love everybody to join the evening reception that begins at 7:30 p.m. So again, thank you for your attention, your time, your profound wisdom, and your support. Have a wonderful evening. Thank you so much.